stare separati.
al margine, però non so come preferite.
No, va bene, va bene.
Va benissimo.
Ci stanno provando.
Cioè, se volete stare insieme,
No.
vedete tutti da lontano. Lo dicevo per voi, ecco.
Okay.
Good morning.
Che bella voce. Do you want water?
Yes, we have a lot of water. Oh my God.
Ritardarla, tipo di 10 minuti, no?
Ma di 5 secondi inizia alle 8:30?
Sì.
Ok. Molto bello, sì. Senza quanti grandi artisti. Per mettere a trance.
Tu lavori qua o?
No, faccio hostess per questo congresso. Quindi è stato forte anche per me.
E abbiamo nell'audience anche Giulia.
Esatto.
Dov'è? Tre, quattro.
No, solo gli amici, ragazzi.
Ma infatti diceva anche il tecnico che ha voluto portare le carte.
E poi ci hanno assegnato questa stanza gigante con anche la seconda, nel senso che questo è proprio per gli artisti. Umiliazione.
No, guarda, si sta bene.
Secondo me abbiamo 5 minuti per
Non vi siete persi anche quel giorno, anche se... Io ce l'avrei domani. Però se...
No, no, ok, lasciamo così.
Scusa, ma cancello. Sì, adesso dobbiamo andare. Poi lo guardo su. Per un po' non
Wired for silence. Silence.
Sì, è una parola difficile.
Caffè su? Però mi piacciono questi microfoni perché sono...
Saranno più o meno 150 questi, no, 150.
vicinissimo non purificando.
Ma quello lì come funziona?
Sì, dove?
Perché Zeniti anche
E te l'ho messo io. Perché prima ero lì vicino. No, scusati. Adesso ricambio sedia, ti confonderei di nuovo. Anzi, se passi per caso lo sposti così mi devo spostare di nuovo.
non ne abbiamo dei movimenti. Casomai se anche tu stessi venendo in sottopunto, adesso vado io così c'è una persona in più.
Sì, certo, è un'ottima idea.
Oppure possiamo le persone che qui.
Ma no, siamo qua. Quando ci rifletto. Guarda qua, guardiamo tutti. Vedi la cascata di Zeniti.
Sì, dai, dottore, possiamo iniziare.
Vale,
Questo c'è
caffè. Il simposio alle 10.
Un altro simposio.
Sì, allora sono tutti amici.
Per l'annuncio.
Senza prenderci 5 euro.
Sì, si può mettere per l'annuncio.
Non esiste. L'ansia.
Good morning. We are going to wait a couple of more minutes before to start to give time to breakfast.
Anche se non penso che in effetti
No.
quando sarebbe il Max ci saranno più persone.
Dove? Se ne andranno addirittura.
No, ci saranno più persone.
Ah, più persone. No, due persone.
Ormai li perdiamo insieme. Quindi paghiamo noi. Oggi viene uno di
Ah, visto? Fatto bene aspettare.
Sono mai visti tagliati.
Sì, esatto, però questo è da mettere, non lo so, in modo che
No,
sia.
perché se togli i primi 4, non è che sono.
Sì, se dai i bravi.
Sì. Posso farmi una foto? Però no, da proprio all'esterno, vedi dove c'erano più. Sì. Penso sia tutti capito infatti tutti questi dove sono stati.
Guardare la cosa che sia.
E che cacchio! Ma ci siete anche voi comunque. Sì.
Ciao mamma. Ciao.
Oh, cos'era? Ok.
È per...
Quando vuoi.
So good morning to the few survivors who made it here today. Welcome to our symposium, The Retina and Beyond, Linking Vision to Behavior. So I'm Serena Riccitelli, a researcher at IRCS Ospedale Policlinico San Martino, and it's with a great pleasure that I chair this session with Dr. Lucia Zanetti from Pisa. She's a postdoctoral researcher at Katia Reinhardt's lab. And our symposium will explore how the retina not only encodes the visual world, but also directly shapes our behavior. And our speakers will present recent findings ranging from new strategies to restore visual function to how the pupil changes how the retina encodes visual stimuli, and finally how retinal outputs are integrated in visual motor pathways. So it's with a great pleasure that I'm presenting the first speaker, Stefano Di Marco. He's an associate professor at the University of Genova, and today he will present his talk entitled Engineering the Retinal Code, Nanostrategies to Restore Vision and Behavior. Stefano, the theater is yours.
Thank you. Okay, thank you. Thanks, Serena and Lucia, for the kind invitation. It is honestly a pleasure to be here with you and with these great speakers. And I would like also to thank the audience for being here so early in the morning. It's honestly appreciated. Okay, today I would like to talk about a new strategy that we are implementing, trying to restore vision, or at least partially vision, in patients that have been affected by degeneration of photoreceptors. For this reason, I would like to start by trying to define with you what is vision. So when I ask this question to my students, they usually reply that is our ability to discriminate the different objects that are in front of us. And so, for example, if we look at this beautiful image, picture from Ansel Adams that was famous for these incredibly sharp landscapes, we can discriminate the stones here, at the bottom in the river, the stones that compose this mountain, the different details of these clouds. But what is interesting is that if you really think that this is actually our main function of the visual system, it is actually performed only by 5 to 8% of the entire retina, while the remaining 95% of peripheral retina is actually encoding movements of objects, animals, or also the movements that we produce while we are actually moving around the world. So it is obvious that this complexity, this spatial organization obviously subtend a very complex retinal circuitry that in fact results in a very complex retinal output. We now know that more than 30 different types of retinal ganglion cell, each one encoding a particular aspect of the visual field with its own particular code. And if you like to try to restore vision, a natural sensation of vision, we actually should aim to try to reproduce this natural diversity of retinal output. So, having this in mind, and as always hoping for the very best, we tried a new strategy by inventing a new compound that we called the Ziapin-2. We wanted this compound to have some particular features. So the first property, we wanted it to be a photoswitchable compound. And this is possible because we based it on the azobenzene chemistry that allows this molecule to isomerase from trans to cis when illuminated with the cyan light. And so does also our molecule. The second important properties we wanted it to have was to be amphipathic, similar to phospholipids in our membrane, and this is why we placed two polar heads here and one hydrophobic chain azopan here. And this amphipathic nature gives this molecule the ability to spontaneously partition within the neuronal membrane. So in this image, you can see a neuron that has been, whose membrane has been labeled with red. In green, you see Ziapin-2, and you can see that they co-localize within the membrane. And once Ziapin-2 is getting within the membrane, we can see, thanks to this molecular dynamic simulation that has been performed by Professor Luca Maragliano at the University of Ancona, we can see Ziapin-2 that can flip on both sides of the membrane and interact with its hydrophobic chain. When this happens, Ziapin-2 will create local invagination of the membrane, thus reducing the thickness and therefore increasing membrane capacitance. Now, when we increase the neuronal membrane capacitance, we are actually making the neuron less responding to synaptic input, slower to respond to synaptic inputs. All of this happens when it's dark. But then we illuminate with the cyan light, and this molecule isomerizes, breaking this hydrophobic interaction within the membrane. And membrane is released back to its original values. And so does also capacitance. And it has been shown from our lab in a previous work that this allows the neuron to quickly hyperpolarize and then depolarize, allowing it to fire action potentials. So this is the reason why we chose this molecule to try a new approach and create a liquid injectable prosthetic device. And we decided to test it on 2 different animal models to show that this molecule is independent from the mutation that caused the degeneration of photoreceptors. And in fact, and also is independent from the species. So if we used a mouse model, RD10, and the Royal College of Sargent rats that are 2 models for retinitis pigmentosa, bearing 2 different mutations that anyway will lead to the total loss of photoreceptors in about 3 months. Given the fact that the results we obtained were very, very similar during the presentation, I will show you results from both these experiments. Just to be sure that the degeneration was complete, we actually selected only animals older than 6 months. So we started with ex vivo experiments. We dissected the retina. Then we bathed the retina for half an hour in oxygenated Ziapin-2 medium, and then we placed it back to a normal medium solution to perform patch clamp experiment. And we interrogated the retina by using 2 different stimuli, cyan light that will isomerize Ziapin-2, so we'll show the Ziapin-2 responses, and a green light that instead, as you see, falls out of the absorption spectrum of Ziapin-2, therefore will not elicit a response in Ziapin-2, but if any residual photoreceptors is there, it could be sensed by this photoreceptor, and therefore we use it as a negative control for the experiments. And these are the results. So when we illuminate with the green light, as you can see, the retinal ganglion cell do not fire any action potential. They don't change actually their spontaneous activity. But when we shine cyan light, we obtain really different responses. So we can see on responses here, but also 2 different types of off responses here with different dynamics. And this was something surprising to us. We were thinking to observe a more stereotyped response. Instead, we can see a real different responses from this retinal ganglion cell. At the bottom, you see a raster plot in which every dot represents an action potential, every line represents a trial of the experiment. And this is good because you can see how very consistent were light responses throughout the entire duration of the experiment. So we now can see light responses from retinal ganglion cells, but our question was, are these light responses originating within the retinal ganglion cell or originating from the reactivation of the entire retinal network? So we performed voltage clamp experiment on retinal ganglion cell, trying to dissect synaptic excitatory input from synaptic inhibitory input. This is possible by performing a conductance analysis. And I remind you that in the retina, especially in the retinal ganglion cell, the excitatory input it's coming from cone bipolar cells, and here it has been represented with a black line, while inhibitory inputs come from amacrine cell. So, and here this inhibitory input is shown with the dashed line. And as you can see, the cyan light was not only able to evoke a spike response that has been recorded previously to get in voltage clamp in a cell-attached mode in this cell, but it's also able to elicit synaptic inputs onto the retinal ganglion cell, meaning that the light responses are derived from a retinal elaboration. So we are actually activating not only retinal ganglion cell, but also the entire retinal network. Another interesting feature of this molecule is that it's also able to work at very low luminances, and this is usually an important point for retinal prosthetic device. We obtain significant responses already at 2 milliwatt per square millimeter. Okay, so now we wanted to try to dissect pharmacologically the source, the origin of our light responses. And the first thing we wanted to really prove was that there was no residual photoreceptor providing any response. So the first thing we did was to block the mGluR6 receptors that allow transmission from a photoreceptor to the on pathway of the retina. And this is blocked by LAP4. And here on the left, you see the responses of a normal sighted animal. This is called RDY. It's the control for Royal College of Sargon rats. And here you see a non-retinal ganglion cell responding to the light stimulus, but you can see that this response is abolished by LAP4. This is an on-off retinal ganglion cell, and again you can see that LAP4 is very specific for the on part of the response, but not for the off. But when we look to our blind animal added with Ziapin-2, you can see that the on responses are resistant to the drug, meaning that these responses are not originating from photoreceptors, but from the retina underneath, so bipolar cells. So now we have in the retina two sets of bipolar cells. We have rod bipolar cells, we have cone bipolar cells. And they actually are interconnected thanks to the A2 amacrine cell. For this reason, we decided to block, by using strychnine, the glycinergic receptors. And this actually induced the reduction in the responses of about 20 to 30%, meaning that the majority of our light responses were actually driven by cone bipolar cells directly, and just a smaller proportion were actually induced by activity in the rod bipolar cells. Finally, we wanted just to be sure that rod bipolar cells were actually needed, cone bipolar cells were actually needed in order to observe a light response, and so we blocked the AMPA kinase receptor by using CNQX. And in fact, this actually abolished light responses in our retinal ganglion cells. So providing a proof that this is actually originating within the cone bipolar cells. So we actually always performed single cell patch experiments. So we were looking at the activity of cells one after the other, but we wanted to see whether we were able really to repristinate this and restore this natural diversity, or at least a diverse coding capabilities in the retina. And we wanted to see that simultaneously. So we exploited the technology of a 3-brain biocam that allows to record from 4,096 electrodes. So when you place a mouse retina on this MEA, you can record hundreds of retinal ganglion cells. And these are the results of the responses we obtain. This is just one example. We have 5 flashes of light, and cells have been sorted here for their polarity of response, so going from on retinal ganglion cell on off and off at the bottom. And you can see that each one of these retinal ganglion cells is actually encoding this light stimulus by using its own code. And we have a real divergence of behavior of these retinal ganglion cells in response to the light, while green light was not able to elicit any response. These are just examples of retinal ganglion cells we are observing. I want just you to focus on cell number 4, which also shows a sort of light adaptation during the stimulation. And this is nice because it shows also that we can reestablish short-term plasticity effects within the retina. And finally, in the pie chart, you can see that we were able to elicit light responses in more than 60% of the retinal ganglion cells that we were able to record with our MEA. The last experiment for ex vivo part, we tested also if our retinal ganglion cells were able to encode the movements, and actually they were able to encode the movements up to 15 hertz. We presented bars drifting at different hertz, and we were able to see ganglion cell firing up to 15 hertz. So Having obtained such a nice results in the ex vivo, we passed it to the in vivo, and what we did was to inject in the intravitreous body this molecule, and we recorded, we tested the light responses in the behavior of these animals up to 2 weeks. We actually chose 2 weeks because after this time, this period, Ziapin-2 starts to be degraded within the membrane, and so we would have seen a quite strong rundown afterwards. So, the first test we performed was a light-dark box in which we exploited the natural aversion to light of rodents. In this arena, you have a transparent part, an opaque part, and we place the animals when it's complete darkness. In green, you see the wild-type normal-sighted animal. In red, you see the blind animal plus Ziapin-2, and in black, you see the blind animal plus the vehicle. And as you can see, in the darkness, they all behave similarly, passing the same amount of time in the two parts of the box, and behaving in the transparent part similarly. But things change when we turn on the light. In fact, as you can see, wild-type animals and Ziapin-2 animals tend to spend the majority of their time in the dark box. And also, when we look at the tracking in the transparent area, you see that the wild-type tends to stay always very close to the walls, as the Ziapin-2 animals, but while the blind animals plus vehicle are actually behaving as if they were in the darkness. And this actually happened for up to the 14th day. Then we measured optomotor response, which is a reflex that is induced by rotating stimuli. To measure this test, to perform this test, we placed the animal in this apparatus that is able to track the head movements, and if the head movements are actually in synchrony with this random rotating stimuli, then the machinery will score a value higher than 1.2. While if the animal is just moving his head in a random way, it will score less than 1.2. And again, you can see that for 14 days, the blind plus vehicle animals were always less than 1.2, while the Ziapin-2 animals actually behaved more similar to wild-type animals. So, in conclusion, I have shown you a new strategy to try to restore vision in patients affected by different forms of retinitis pigmentosa or other photoreceptor degeneration. This molecule is able to reestablish a complex coding within the retina by emerging on and off responses. It's able to do this by reactivating actually the entire retinal network. It has been proven to be functional also for in vivo. I didn't really have time, I'm sorry, to show you biocompatibility and safety results, but you can download the paper using this QR code and the data are there. The paper has been published at the starting of this year. And this molecule can also reestablish motion analysis. And finally, for the future, what we are trying to do is to try to increase the amount of time that Ziapin-2 can stay within the retina. So with this, I would like to thank the group, the entire department. We are almost a community, and this is a beautiful picture because you can see not only the teammates, but also their family, our family. And I think it's very nice because we all know that if we are here and we can do actually this job, it's just also because we have our family behind. So thank to them and thank you for your patience.
So thank you, Stefano, for this fascinating presentation that gives us some hope. So are there any questions from the audience?
I could start if there's one.
You had this in your slide, you mentioned that you're looking at how frequently you'll have to deliver Ziapin-2. So do you have any idea about how often you'll have to do that, and do you have any strategies to try to increase the half-life of Ziapin-2?
Yeah, thank you for the question. Yes, so unfortunately, Ziapin-2 will last within the retina up to 2 weeks. We are sure of this, but after 2 weeks start to be degraded. So we have a few ideas. One idea that it didn't really work was to try to increase the number of polar heads to try to make it anchor more on the membrane. It's not really working very well. This diapin-2 is working better than the other molecules. We are trying to embed it in microplatelets in order for a slow release within the retina. And we are also trying to avoid the MSO by placing this molecule within the liposomes to try to reduce the toxicity and increase concentrations. So these are actually the strategy we are trying to implement for this. But
Your methods will make all cone bipolar cells on bipolar cells, correct?
we think that the majority of the responses are probably driven from on-cone bipolar cells. We don't have experiments really to prove this. We are actually working on that. But we think, we have an idea that it's where it's deriving because on bipolar cells are longer and this capacitive effect probably there are more effective in blocking during darkness any activity. And so when you turn on light, we think that yes, it's by probably moving more the on bipolar cell that we obtain the responses.
But even if they activate, if they modulate the off bipolar cells, they will also become on bipolar cells.
Yeah, but so this was actually why we were thinking to observe a stereotyped response there. But as you can see, didn't happen. And we think that it's probably due to the fact that given that the membrane of these two neurons and the surfaces are a bit different, probably the effects are different, but we cannot probably say that are null in one bipolar cell and complete in the other. So we actually are trying to investigate why we can observe such a great diversity. We think that it's mainly because we actually are reactivating the entire network, and maybe also that serial inhibition can activate off responses by disinhibiting. So if you activate on a macroin cell, you can still disinhibit off. So these are ideas, but we have to prove this.
Can I add just one question according to that? So, like you know that I like the direction selective ganglion cells and the circuit is very complicated. So, like I want to understand whether when you speak about encoding movement, did you really see direction selective ganglion cells? Because this would be like a super pro or just spatial frequencies?
No, we haven't really checked for direction selective retinal ganglion cells. We just checked whether retinal ganglion cells were actually able to encode, track these movements. But this is another thing, so we would like to see and classify better retinal ganglion cell. Another thing that I didn't really say is that on cells, on retinal ganglion cells that looked on morphologically were also showing on responses. Off retinal ganglion cells were showing off responses. So it looks like the retinal circuitry is activated, and maybe that direction selective also are activated.
So thank you, Stefano.
Thank you.
We can move to our next speaker, Dr. Santiago Rompani, a group leader at MBL in Roma. And today he will present his talk entitled Pupil Modulation Drives Retinal Activity in Mice and Human Perception.
Hello. Okay, very good. I have the clicker here. Thank you, everybody, for joining today. Today, I'm going to be talking about the pupil. It is a structure in the outer eye that can dilate and constrict in order to allow more or less light in from the environment to assist the photoreceptors. The rods and the cones, they can actually have a dramatic ability to adapt to different light levels, all the way from the dimmest starlight to bright sunlight in a cloudless day. Now, the issue with this is that this dramatically large dynamic range, one of the largest in nature in terms of being able to be very sensitive at various different domains of intensity, has a problem of speed, which the pupil should help. Because the pupil, at its maximum dilation and constriction in various species, can only really go to shaping about 1 log unit of light. So not that much in terms of what the photoreceptors alone can do. And even in an evenly illuminated room, there is huge dramatic differences in the intensities between the darkest shadows and the brightest lights that can usually be a 100-fold difference, meaning that the pupil's modulation is actually, in fact, quite narrow. However, this can be very actively seen when you go from, say, a very bright sunny day into a darker structure, like this beautiful cupola that you actually can't appreciate, but then your pupil dilates in order to let more light in, so that as the photoreceptors are becoming more sensitive, they're being helped by a larger flux of light. Now, the pupil is thought to have other roles as well, but this is considered to be the main one, where it's helping these photoreceptor cells in this process of adaptation, and basically by definition for this function, they should not be stimulating the ganglion cells, the cells in the retina that project from the retina to the brain and largely encode various different features of the visual scene. The pupil kinetics, in fact, is supposed to help blunt a lot of the effects of dramatic light changes to allow the retinal circuit to do its particular computations. This has allowed the various fields to use the pupil constriction and dilation to read out arousal, because in fact the pupil is driven by a circuit that is very influenced by the activity of other brain circuits that largely report arousal, namely the locus ceruleus. So you can just look at a pupil constricting and dilating in an evenly lit environment and infer the arousal state of the animal with reasonable certainty that it's not affecting vision, right? 100-fold difference, whereas the pupil is only doing 10x. This, however, is an assumption, because oftentimes, as our previous speaker noted, people do a whole mount preparation of the retina where there's no pupil anymore. So the active ability of the pupil to shape ganglion cell responses has not been directly looked at. And this is something that my students Tiasha and Pietro sought to pursue by injecting an AAV into the eye that in only ganglion cells, using a cell type specific promoter, expresses GCAMP, either 6S or 8F, and we image from the ganglion cell axons in the lateral geniculate nucleus of the thalamus, the part of the thalamus that receives retinal input and projects to the visual cortex. And this, here you can see the craniotomy as well as the labeled cells. We only inject in one eye, the other eye being unlabeled. And here you can see a brief movie. Oh, the movie works, that's nice. With the activity of these boutons in vivo in the head-fixed animal. Now, so the experiment is as such, where Tiasha and Pietro have two screens, one simulating the eye contralateral to the surgical site, which is where we injected the virus, or another screen in the opposite ipsilateral eye. And if there is no pupil-induced activity, as the previous model would suggest, you have, say, a full field flash, where you have a gray screen that then becomes brighter, and then goes to gray again. The pupil will constrict. It'll go from this dilation in the left to a constriction on the right, but the detected stimulus by ganglion cells will still be just a square pulse of a full field flash. So if you look at off cells, for example, cells that, ganglion cells that fire when the light turns off, you will see activity here, where the flash of light goes off, the canonical off response. And in fact, in this example Bhutan, this happened, where you stimulate the injected eye, where you saw activity, and if you stimulate the opposite eye, nothing happens, right? Those are not labeled ganglion cells, nothing is happening. The eye that is injected is just receiving a gray screen. Now, in contrast, if the pupil can shape this full field flash, you receive the flash, the pupil constricts, meaning that there's a bit of dip in the intensity of light, meaning that that same off cell now has another stimulus, here in the red arrow, a pupil-induced monocular response from the same eye. Interestingly, also, if you stimulate the opposite eye, the pupil is consensual, in that stimulating one eye, you get both pupils constricting, because there's a quick circuit from ganglion cells to two subcortical regions that then go right back into the outer structures of the eye to constrict or dilate the pupil. So stimulating the opposite eye will induce pupil constriction, and since the screen is gray, it'll actually become dimmer as well as this. So we would actually expect to see activity. And this would mean that that ganglion cell axon that is in the brain is effectively binocular, because a binocular cell is a cell that responds to stimulation of either eye. And in fact, here's an example Bhutan seen by Tiash and Pietro, where you can see just what we predicted. A pupil-induced monocular response in the left and a binocular response in the right. Now, is this correlated with the pupil? It is. You can see here that this monocular and binocular response correlates nicely to the kinetics of how the pupil closes in the mice as a population level. And if you look at the single ganglion cells, of which we have a few hundred here, you can see that the monocular and binocular stimulus on the top is present in a large plurality of these recordings. And in the bottom, we actually did the causal perturbation to see if the pupil is actually causing this by eye drops that create a hyperdilation of the pupil, preventing it from constricting. And if you do this, these putatively pupil-induced responses are removed, but the canonical off response is maintained. In addition, if you inactivate the ipsilateral eye with TTX, this should prevent the consensual pupil constriction, getting rid of the binocular, but not the monocular pupil-induced response, and in fact, that's what we see, suggesting that these are not just correlating with the pupil, but in fact caused by the pupil, and the TTX control suggests that it's not due to some light contamination artifact, either through the stimulation apparatus or within the skull. Now, this was off cells. Does this happen in all the ganglion cell types? For this, we used the chirp stimulus, which is a series of flashes changing in different frequencies and amplitudes that generally catches a large plurality of the ganglion cell types in the retina. And we were able to cluster this into several populations, but for simplicity, we just went with 5 very simple ones, of which 3 are off cells and 2 were on cells. And we can see some monocular modulation, which I'm not showing, but very dramatically, you can see the binocular responses from the ipsilateral stimulation shown with the red arrowheads. And the on and the off cells, if you look at just their ipsilateral responses, they cluster into 2 discrete groups, the ones for the off ganglion cells and the on ganglion cells, that largely behave as you'd expect, with the off cells responding to the decrease in the pupil size with an activity, and the on cells with a decrease with some kinetics of oscillation that happens afterwards as well, in opposite polarity between the 2 cell types, consistent with their known functions. Okay, so this is in a lot of different cells. The amplitude of these responses are comparable to that of canonical responses. The question now is, what is the function of this activity? What is it likely doing for the organism? Is this some evolutionary artifact, or does it actually provide some encoding function? So to look at that, we actually found something about what binocularity does. Because the main role of binocularity in most species is seamless depth perception, stereopsis. You're able to see if something feels closer or further away very quickly. If you close one eye, you can kind of sort of tell, but it's very slow. Trying to catch a ball moving at your face with one eye closed is very difficult. However, there's another function of binocularity, which is binocular summation, where you actually have much higher resolution in various categories of vision using both eyes compared to just your dominant eye. And this is something that we probed for by stimulating just the contralateral labeled eye and comparing it to both eyes. For this off cell, for instance, it's known that if you stimulate both eyes, the pupil constriction is stronger than if you stimulate just one eye. So you get the pupil facilitating, where there's more of a pupillary effect. And we asked, would this lead to a facilitation of that pupil-induced response? Is there a binocular facilitation of this response? And in fact, we did see this, but surprisingly, we saw it most for low contrast. So if you start from a gray screen and it becomes just slightly brighter compared to a gray screen to a much brighter screen, which was a bit surprised. This is statistically significant. The top is an example, the bottom is, so you can see this over hundreds of boutons, particularly because the pupil change in low contrast is much smaller than in high contrast, and the difference between both eye and contralateral eye is actually quite small, but that seems to lead to a very dramatic potentiation, as you can see in the red arrow. And if we add atropine, the effect goes away entirely, just as a control for that. You can see here this quantified by the correlation between the pupil constriction and the neuronal activity in a bouton by bouton level, and seeing that this goes away with atropine, with the effect, of course, being very substantial at low contrast. And importantly, if you actually take the response, the individual response of stimulating each eye separately, at low contrast, that is less than stimulating both eyes at once in 40% of the boutons, meaning that this is a more than additive effect, a facilitation that is substantial, despite the low amplitude of difference in how the pupil closes or opens, specifically in low contrast. Now, this low contrast is a bit exciting because we currently live in a very high contrast world. We have stop signs that we really want to see while we're driving cars. But in nature, in fact, a lot of the most salient stimuli are low contrast. It's the tiger hiding in the grass or the fruit that is like hidden behind leaves. So the fact that this domain of vision might allow for potentiation of low contrast stimuli has possibility for having a real ethological role. More about this soon. In particular, if this gets relayed to the cortex and encoded there as well, because there are other domains of vision, such as saccades, where you move your eyes constantly, but you don't perceive that, because a motor copy by corollary discharge is used to inhibit the strong activation of the retina when your saccades are moving. So we asked, is something similar happening here? Because there's also a motor copy that the brain can use to just average this out. It's present in ganglion cells, maybe it's gone in cortex. Long story short, we imaged the thalamic axons in V1, as well as the layer 2-3 cells in V1, and it is maintained. We don't see it as strongly, of course, because the cortex is not well excited by full field stimuli, as was initially shown a long time ago by Hugo Wiesel and even earlier. But we do see this pupil-induced response quite significantly already in the cortex, which brought in the extra tantalizing question of, is this conserved in humans? We actually expected not. Mice are very nocturnal animals. Binocular facilitation in nocturnal animals is more prevalent because presumably they want to operate in the lower contrast world of more complete darkness. And human fovea is very optimized for these sharp edges and higher frequencies, which this pupil modulation wouldn't happen with. And for other reasons of just, the systems can be perhaps quite different. A big difference is that this pupil constriction takes 1 second to happen in the mouse. That is a derangedly long amount of time in terms of vision. You can recognize faces and report it in less than 200 milliseconds, usually less than 100. 1 second is a domain of vision that is qualitatively different than most vision that people have studied and that look at in terms of our immediate responses to stimuli. However, collaborators we have in Helsinki replicated our stimuli exactly in humans as we did in the mice, and they find that that delay is actually also maintained in the humans. Furthermore, if we match stimuli to the exact pupillary kinetics, in details that I'm not going to discuss, that is also very tightly conserved between the mouse and the human, suggesting that our study, that what we find in mice could actually be replicated in human perception as well. So to do that, Tiasha and Pietro devised this really clever psychophysical experiment where they just asked a volunteer to report if they see when the screen gets flashed, if they see this dimming happening right after, that would correlate with the pupil constricting. Here you see the results in 2 volunteers, one at low contrast, one at high. One volunteering seeing that dimming at low contrast but not high, and another volunteer seeing in both conditions, both correlating with the pupil at about 1 second. Again, far slower than you'd expect from normal reaction time for vision. And if you look at our 8 volunteers, you can see that there is a lot more detection at low contrast than high, like we saw in the mouse, also despite there being lesser pupillary constriction at low contrast than high. So in conclusion, this is the result of the paper that the lab produced on this, our first paper, where we show that in addition to cortical binocularity, there is also this pupillary binocularity that occurs already in the retina, that this likely helps with low contrast binocular facilitation, that this is conserved between mice and humans, and that it is actively perceived. Now, currently, we're continuing this work to look at how pupil shapes vision during naturalistic stimuli. For instance, with this movie of a view from a train, or something like this one-take shot from an early Orson Welles movie, in order to combine that with not just pupil size and locomotion, but also face map, in order to combine the neuronal activity with behavioral regressors to really look at how the pupil interacts with internal states and its locomotion and size, as well as constriction and dilation. We have some preliminary data from layer 4 cortical cells for this Touch of Evil movie, where we can see a very stereotyped response of these populations, and that now we're looking at how the pupil's activity itself is shaping these very contextual responses in the visual cortex. So I'd like to end by thanking the people who did the work, and of course, all of you for listening. Thank you.
Thank you, Santiago. Are there any questions from the audience?
Santiago, the running is correlated with pupil size changes, and it has been shown in visual cortex that somehow, that running itself modulates it very strongly. But your data is suggestive that pupil size luminance changes will to some degree be part of it. And I wonder, how much do you think it is? Could it explain it all?
That's a very good question, and in fact gets to the heart of how people would be interested in this in the context of the locus ceruleus that drives a pupil is also a huge locus of arousing the brain, and how are these two interlinked? I think it plays a role, but certainly not all the role. But even in ganglion cell axons, people reported like locomotion having this modulatory effect on activity, like the Schrödinger and Liang papers. And the controls they did to rule out that this is an effect of vision, I think, are flawed, given these findings that we made. We can talk a bit more in detail on that. So this is why we're doing these net subsequent work, and why we're including so many other domains of recordings, to see if we can parse out what is visual retinal activity and what is perhaps other domains of internal states separate from arousal. But interestingly, yeah, pupil and locomotion are correlated, but not perfectly correlated. So there are domains in which they differ in terms of how they drive neuronal activity, which is an interesting part. And anyway, we'll just do a bunch of this stuff with and without atropine and carbachol, which will help us dissect a lot of this out, even though they add some artifacts there that we would want to do some other things to control for, to really say quantitatively what is the effect of the vision being shaped by pupil in these effects. I think there was another question there.
Thank you so much. So I'm not so familiar with pupil, but I was wondering what's the mechanism, so what's the connection between pupil and retina ganglion cell, and so which type of synaptic connection there are between these two structures?
Okay, so there's no synaptic there's no synaptic connection, The pupil is just an opening, like a shutter in a camera, letting more or less light in, and it's affecting the retinal circuit, photoreceptor to bipolar to ganglion cell, in order to shape what the ganglion cell is seeing. So without a synapse, it's able to affect this. The ganglion cells themselves project to certain brain areas that then in turn affect the pupil. So if you see a pupillary reflect, it's because there was a retinal input driving it, but the activity of the pupil onto the retina is non-synaptic.
Okay, thank you.
Pleasure.
Thank you for the presentation. Have you thought about the physical effects of light? So you mentioned the fact that the quantity of light, of course, is different, but also the quality of light. And think about the nearby vision. You have the more spread light rays, while on far distance you have this, physically speaking, parallel light rays. It could also help in this contrast and nearby vision. For example, we humans have the convergence mechanism that has also this.
That's a good point. They're not in terms of polarity, like that's polarization that happens in very far, but there is depth of field.
Yeah.
Yeah, I think that's what you're mentioning, yeah.
Exactly.
Where for sure the pupil big versus small has a different depth of field effect. That's very important in humans, not really in the mice. So much for various reasons. Its visual system is not as peak as human. So yes, that interaction is important. The effect that we're seeing happens when you have the actual closing and opening, not at the biggest and smallest state. If this were a depth of field effect, we would have with atropine, very big, carbuncle, very small, different. We only see this effect when it's a transition between the states. If we do these two perturbations, there's none of these inductions. So we don't think it's related to depth of field in the mouse. We're still doing more human psychophysics to tease it out more there. That's a good question, yeah.
Thank you very much, Santiago.
My pleasure, thank you.
So we move to our next speaker, Dr. Norma Kuhn. She's a postdoctoral fellow at the lab of Karl Farrow at NERF Leuven. And today she will speak about wired for saliency, visual feature processing in the superior colliculus.
Hi everyone, and thank you Lucia and Serena for inviting me to give this talk here. Very exciting meeting. And this talk will focus on something completely different, as I'm really interested in how the spatial setup of a neural circuit or central neural circuit actually helps the processing of information that's important for the animal when it forages in the environment. For instance, to look for food, like here this, sorry. Okay. So imagine to be this little mouse, and you look in the wild for food, but as you're looking for food, you also always need to be vigilant about predators up in the sky. This means that this animal has to very quickly process all what is around it in a highly efficient and fast manner. And this talk concerns how the spatial setup of the circuit of the superior colliculus actually helps and facilitates this process of fast detection of these behaviorally relevant stimuli. So as I already said, the circuits that I'm interested in are the circuits of the superior colliculus, which transform visual stimuli into innate orienting behaviors. These, for instance, could be these freezing behaviors or also pursuit behaviors of prey. But how often when we look at these transformations, we look this by choosing networks of very simplified neurons. However, as we all know, neurons have these beautiful ramifications called dendrites, along which they integrate. There are thousands of inputs. And these inputs are not also just at random, but they're also highly spatially organized. For this And what I'm trying to show you here, that actually it's the spatial organization of these dendrites and their inputs that facilitate the processing of behaviorally relevant features. And I look at this by looking at the wide-field neurons in the superior colliculus. And the colliculus receives direct input from the retina, so direct visual input. And this input is first of all stratified, so specific features like direction and contrast arrive at different layers of the colliculus. And also retinotopy of the colliculus ensures that space in the visual environment is translated into anatomical space, which means we can integrate spatial information, especially in these beautiful wide-field neurons here. So the advantage of looking at these wide-field neurons is that they really have a dendritic tree that spans the entire depth of the retinorecipient layers of the colliculus, so it can integrate all these different inputs and also spans a wide space of the visual field. And these neurons are very important, not only for freezing in response to an approaching predator, but also for pursuit behaviors and detecting prey. And also because we have these different inputs that arrive at different places, these dendrites allow different computations. So one of them is bandpass filtering, another one are nonlinear operations, and also spatiotemporal summation of the different inputs. And the strategy to understand how the dendrites help in processing salient information is first we need to know what the inputs are. So we will trace the inputs directly back to the retina and record from the retina. Also, we need to know what the outputs are. So we will record from these wide-field neurons in response to visual stimuli. And then once we have the inputs and the outputs, we can determine the transfer function, which means we can use different computational models to understand what are the important features of the dendrites that help transforming the retina inputs into outputs. And we started by first measuring the responses of the wide-field neurons. Here, fortunately, we have a genetic mouse line, which allows us to encode a calcium sensor only in wide-field neurons and measure the responses in a head-fixed setup by implanting a window on top of the superior colliculus. And here you can see such a recording. So here you have the response of wide-field neurons while the animal is head-fixed and seeing these looming stimulus. So this is one of the stimuli that we present. And you also can see a short stopping in response to the stimulus. Here's an example recording where I hope you can appreciate that actually these wide-field neurons have a very broad response properties. So they are differing in their responses, but also they respond to a very broad set of stimuli. And we can group these stimuli into 3 groups, sorted by their contrast preference. So we have on cells that respond to bright stimuli, off cells that respond to dark stimuli, and on-off cells that respond to both. And the stimuli we presented here are either approaching objects, then receding objects, which is, will become important for the rest of the talk. Then a dimming stimulus, which doesn't have a moving edge. And I hope you can appreciate here that all of the cells, they don't respond to this dimming stimulus. So moving edge is very important for these cells. That's why we also call them multiplexers of motion cues, because they are responding in general to a lot of different ranges of motion. And we also looked at the dendrites. Here we looked at different depths. So we measured response in the very distal dendrites up down to the soma. And here we use the full field stimulus, to which we don't get a strong response in the somas, but we can nicely see that different parts of the dendrites will show different types of local responses. And again, when we cluster this, we can see that there is an organized, there is a layer-specific organization to these responses in the dendrites that we see, where, for instance, this cluster is primarily in the most superficial layers, whereas these type of responses we only see in the deepest layers of the colliculus. And this can happen really within the same neuron. Okay, so now we've classified the outputs of these neurons. Now let's have a look at the inputs. Here we use the rabies labeling strategy, which allows us to exclusively label the retinal ganglion cells that project to white-filled neurons, not the other cells. And we record their responses using either patch clamp recordings, but also calcium imaging again. And in the end, we transform everything into calcium responses. And for a subset of the cells, we also recovered their morphology to classify them as well as molecular markers. And here you can see that we actually get a very large amount of different retinal ganglion cell types that project to white-filled neurons, also given that they really span this entire range. And we could identify 12 functional types, where some of them are off cells responding especially to a darkening of the screen, and others are on cells responding to a brightening of the screen. And we could also assign those to known cell types in the retina. Now let's have a look whether we can recover these signals actually in the dendrites of the white-filled neurons. So what we did here, we matched the signals that we measure in the retina with the signals that we measure in the dendrites of the white-filled neurons. And when we do this, we also assign where we find those signals in the dendrites. And this allowed us to draw this beautiful map of layers where each of the inputs arrive. And this also matches well with already known stratification. For instance, the F-mini off cell is known to stratify in the more superficial layers of the colliculus, whereas others like the transient on alpha cells are on the deeper layers. And also let us recover stratification profiles of other so far unknown cell types. Okay, but here we've only looked at dendrites. Of course, we want to know how the white-filled neurons transform the inputs from the retina into meaningful stimuli. So in the first case, we just looked at what happens when we just sum all the retinal signals that we saw, and can we recover the signals that we see in the white-filled neurons. When we do that, we can recover some of the signals. So here, for instance, you get a very nice match of this response to a white looming stimulus and the shrinking stimulus. However, for these off cells, we see that we don't get a very good match for these receding stimuli. And we used a different way to quantify this by looking only at the peak difference. And when we do this, we can see that for this shrinking stimulus, we always underestimate the peak responses. So this means that if we only take the sum of the retinal responses, we are not able to get the same strong responses to a shrinking stimulus that we see in the white-filled neurons. And we also quantified this in another way, where we looked at the expansion selectivity. So in the retina, here in light gray, you can see that we have a very strong bias to looming stimuli, whereas the white-filled neurons respond equally strong to looming and shrinking stimuli. So they really don't preference an approaching thing versus a running away thing, but encoded in a similar way. And now the question, of course, is how can we get this response out of the retinal responses? In the following, I will use this kind of representation where it's the average of all the response of all the white-filled neurons and the fits. And here you can very nicely see that we don't get a good match of the responses to the shrinking stimulus. And the first thing that we did was looking at nonlinear effects of dendritic processing, which are very well studied in the cortex. And so we used a nonlinear multi-layer model that integrated these retinal inputs and passed them through nonlinearities. And we did this with different model complexities. So the very first one is just a one-layer model where we only have the rectifier. Here we don't see much of a difference. When we add a second layer, we also don't, we get a little bit of improvement, but not much. And then with the third layer, we see more improvement. But you see that there is still quite a discrepancy here. And that's also apparent here in the peak differences. So it doesn't, so nonlinearities seem to help, but they come at a very big cost of complexity of the model. So we looked at other things of dendrites that we haven't looked at yet, namely the linear properties of dendrites. So as I already showed, wide-field neurons have these huge dendritic arbors, which means they're most likely low-pass filters. This means when a signal arrives at the distal tip, it will be delayed when it arrives at the soma. But also because we have retinotropy, so inputs that arrive at different parts of the dendritic tree are also linked to a different location space. And since we have a looming and shrinking stimulus, these inputs are not happening all at the same time, but they are integrated over space and time. And so we try to use these two assumptions in two separate models. One is addressing the low-pass filtering of the dendrites, so introducing this temporal delay. And here we suddenly see that we actually get a very good match of these responses here, just by adding a small temporal delay. And the other model is this spatial arrangement model, so that different inputs are activated at different time points. This we simulated by using a filter that simulates this quadratic increase of the inputs. And when we do that, we actually also get a very nice fit of these responses. And if we look now again at the peak differences, we see that the spatial arrangement model really has the best performance. And for the low-pass filtering model, we see that only low-pass filter models that induce a very large delay of almost a second can actually get the same performance as the spatial arrangement model. And this 1 second delay of the signal is not really physiological, so it's not a property of the dendrites, but it actually is very well in line with the dynamics of our stimulus. Because what I didn't tell you yet is that the stimulus takes a second to expand or to shrink. So this actually fits very well with this assumption. But to further test this, we now showed the same stimuli, but with different speeds. So now, so this stimulus is the one that we usually used in this darker gray, which takes a second to expand. We also use stimuli that take 2 seconds to expand, like this light gray one here, or are faster. And as you can already see, the responses to the shrinking stimulus get even higher when we have slower dynamics of the stimulus. And so when we now look at our models again, in gray is the very first model with just the summation of the retinal signal. We can see that it gets worse and worse the slower the stimulus gets. But our spatial arrangement model really performs well across all the different stimulus speeds, which shows us that this is really the underlying structure that is important for capturing the responses of wide-field neurons. And what even more, because I showed you that we have this nice layered input structure of the retinal inputs, what we now did, we only constrained the inputs that arrive at the distal layers to get this spatial integration. All the inputs that arrive at close to the soma, we don't integrate. So this means we even now cut down the parameters of the model. And when we do that, we again get a very good fit. But what it means that we now reduce the inputs, the amount of wires that we need to deliver these inputs. So as a summary, I hope I've convinced you that it's actually the spatial organization of the dendrites and the inputs that forms the neurosubstrate for extracting these different salient motion cues. And so by integrating these, we can then simultaneously track predators and prey. And also that the layer-specific input organization reduces wiring costs without compromising the computational power of wide-field neurons. And if you want to read more about this, the paper just came out recently. And with this, I want to thank the recent and former members of the Ferrolab, and especially Chen Li and Katja Reinhardt, who did all the retinal tracing and recordings, and 2 of my students who helped with computational modeling. And with this, I want to thank you for your attention.
Thank you very much, Norma. Do we have any questions? Thanks.
Thank you for this very interesting talk. You have shown us that the dendritic organization, so the spatial arrangement of the dendrites, is fundamental for understanding the responses of the superior colliculus. And at the very beginning of your slides, you mentioned innate behaviors. So I wonder whether, what are you referring to when you talk about innate, what kind of patterns, and whether you think that these patterns are somehow structured in the dendritic organization itself?
By patterns, you mean stimuli or you mean motion patterns?
Either motion patterns or stimuli. When I think about patterns of stimuli, for example, I think about faces. I mean, I'm biased because I work with human newborns, and there is this long-standing debate about whether face perception is innate or not, and this is why this came into my mind. But this also with motion, it would be the same thing.
I mean, like, so here we really present a very simple stimuli, which are really just a black or a white dot, right, that are moving in different speeds, and like we have translation, but also the approaching and receding stimulus. And what we saw is that these white phase neurons are really just very good at capturing motion of any sort and drawing the animal. So how we think about them is really that they draw the animal's attention to something that's moving. And that can be prey, that can be predator, but it's important for the animal to capture this kind of motion. And particularly these cells seem to be very good at capturing slower motion, which might make me think that actually they're even more important for prey capture than for escaping from predators, for instance.
And the question was, where do you think this, I mean, to capture this motion, for example, the organization of the network should be in such a way that they are particularly sensitive to this kind of motion. Do you think that this is in the dendritic organization or somewhere else?
So what I think is really that it's in the dendritic organization. What other people showed as well, that these neurons, when you stimulate only one distal tip, you can already elicit a spike in these neurons. So this means they're very sensitive to their distal tip inputs, which means they're very good in integrating the activation of each of these different dendritic compartments, right? So this means they will be able to follow a motion that is going across the dendritic tree of these neurons.
Okay, thank you very much.
Thanks. We need to move on. Thank you, Norma. Okay. Our next speaker, Maximilian Josch, is an assistant professor at the Institute of Science and Technology in Austria. And today's talk is titled Intrinsic Dynamic of Visual Attention, the Early Visual System.
First of all, thanks, Lucia and Serena, for organizing this. And thanks for all of you for being here. I'm going to go a step further from the retina and trying to see how actually this information is being modulated by attentional processes. And so let me see if this works. So what is attention? Attention is the taking possession of the mind in clear and vivid form of one out of what seems several simultaneously possible objects of traits of thought. I mean, that was formulated in the 1800s, and it's what's happening right now because you're focusing hopefully on me and not on your phone. And the fact that we can focus on some things, it's not a trivial task. And sometimes you go, for example, when I became a father, suddenly I saw astrologers everywhere, and suddenly I thought the appearance of astrologers was something that happened at the moment, but they were there all the time. So there are objects and features and stimuli that come into my eye, they come into my retina, but I'm not perceiving them as vivid as in certain circumstances, in certain states on others. And I want to start with this optical illusion. And if you look at the center to this green dot, you will hopefully perceive that sometimes you see the yellow, very salient dots, and sometimes you don't see them. So even though those dots are there and they will elicit your activation on your retina, they will be perceived in some instances they will not. And so we were basically curious to try to understand how certain modulations will change the way we actually process visual information. And so this talk is not about what the eye tells the retina, but it's much more about what the eye thinks the retina is telling it, okay? So let me start by telling you who did the work. Florian did most of the training paradigms and two-photon imaging. Victor was fundamental in trying to understand the process and the dynamics that we saw with Anton Sunzer. And Abir and Gasper were part of the modeling of it, which I won't have time to explain today because of time reasons. So what are we looking at? We're looking at the early visual system, and it's basically thanks to Norma. She already introduced the superior colliculus. It's a retinal recipient layer that gets direct input from the eye. And it's organized in three roughly layers. You can think the visual layers, which we were just discussing, then you have a multisensory layer and a motor output. And the idea of this is that the superior colliculus is fundamental in driving certain innate behaviors. It's important in primates to move the gaze, your eyes, and for example, in driving escape behaviors to threats. And another feature of the superior colliculus is that it's retinotopically organized. So classically, what you see is that when you present stimulus in different parts of the space, and this is basically real data, I just encoded each cell as a dot and put the delta change of calcium signals in changes in the intensity of the stimulus. And you see that there are very specific responses that are retinotopical organized. And what you also see is there is nothing else happening, okay? So the idea of this is that the superior colliculus functions somehow as a relay scenario. But that's not how we see. We don't see on the average. We see actually on a moment-to-moment basis. And if you look at the moment-to-moment, what you see in certain instances is that there's a lot of noise. And so to be able to understand what's happening in your surround, you need to either compensate for that noise, ignore it, or perhaps that type of noise is something fundamental in how you interact with the visual world. And so given that we start seeing this type of information, we thought, oh, perhaps it is. And we would like to understand how it is when you're trying to change the state of the animal and perceiving certain very simple stimulus, if that modulates the way, in a causal manner, the way this noise is represented in the superior colliculus. And so what Florian did, he started training animals in this operant conditioning task, and basically it's a detection task, very simple. The animal responds by looking at the appearance of a dot. And what you see on the right, let me see if this works. Oh, there. What you see here is the training. So you see that after a few sessions, the animal learns, their false positive rate goes down, and basically the response rate goes up. So they have a lot, the precision is very high. This is basically depicted here, and most of what you're going to show you next is comparing naive before the training, beginners, and experts, which are basically at that stage of the training paradigm. And to show that this has something to do with spatial attention, if you basically have it predictable, the reaction times are much faster than if you randomize the position of the dots, saying that the animal needs to have some attentional process to be able to lick and as quickly as possible. And so let's take a look at the visual responsive cells. And this is very easy. We can basically take the average, make a clustering, and now we're just taking the black cells here, which are responsive to this visual stimulus appearing at the retinotopic part of the superior colliculus. And we take a naive animal, and what you see over here is you see, let's say, repetitions. This is one example cell, second example cell, third example cell. And these are the visual responsive properties of that cell. And here we take the average. And over here, we have the average over different animals. So you have basically what you normally see. Some cells are on, some cells are more off, and they have the response properties that you depict. When you start training the animal, the response property starts changing slightly. So what you see here is that the response gets increased, and there's kind of like an after effect, like activity in some of the instances. And now take a look at what happens when the animal is trained. When the animal is trained, and it's an expert animal, the responses are much more reliable, way bigger than what you would expect from a naive animal. And the other intriguing thing is that before the stimulus, you have a ton of noise appearing. A little bit of the noise that I was telling you before. So it seems to be that the animal is learning something about the statistical stimulus, so the easiest way of discriminating between is it something you learned or is it a change of state is just removing the spout. The animal immediately understands that there is no stimulus that they will get, and that is basically, it's not necessary to pay attention to the visual environment because I'm not so interested in vision anyway. And what you see immediately after removing the spout is that the activity patterns go back to very much to the naive state, and this noise that I was telling you about before gets strongly diminished. So we found this very interesting, and just for control, it has nothing to do with the behavior of licking. It has nothing to do with the reward. It has nothing to do with differences in running or differences in pupil size, okay? It is very much a stage-dependent change that that is reflected in the activity of the superior colliculus. And so here's a conundrum. Basically, before the stimulus, and this is the coefficient of variation, which basically tells you how noisy the system is. Before the stimulus, in the pre-stimulus time, experts are much more noisy. The activity of the brain is much more active. Whereas when the stimulus comes, it's much more reliable to detect the stimulus. And this is kind of strange. Normally you think in an attentive state you would like to reduce the noise such that the stimulus gets enhanced, but in this case it's kind of like both at the same time. You make it more noisy somehow, and you are better in detecting the stimulus. So we were interested in looking at this a little bit more in detail, and we started looking at, let me see, the spatial properties of what happens in the brain when the animals are across these different states. And this is one example animal. And what you can see here, this is the superior colliculus, the spatial properties of the superior colliculus. And in the naive animals, you see basically very little of these activity patterns. Some animals have more, some animals have less. When the animal starts learning, it becomes more. And at the expert state, this is full. You see basically kind of as if the superior colliculus would be boiling around. So we started looking at this, and since normally if you think of visual sensory neuroscience, you have a stimulus time, and you take the average, just as I was presenting you before. But that's very difficult here, because that intrinsic activity seems to be somehow random. It appears at any position in space. So we need to define a strategy to be oblivious of where the stimulus is, but still being able to determine when this intrinsic activity, these patterns of intrinsic activity arises. And so let me think of it, let's think of it in this example to guide you through the analysis. This is the average visual response, okay? We know where it is, but we will be oblivious of where it is. And how can we do that? We can basically align each cell based on the strongest response. And in this case, we have here 100 cells. Normally we record like 2,000, 3,000 cells. And basically, these are the visual responsive cells. Now there is a metric. It's called population entropy. That we can use, it's the yellow curve here, which tells you something about how the activity patterns are distributed across the superior colliculus. When it goes down, it means that they're skewed. It doesn't tell you anything of where the activity patterns is, but it tells you something about this distribution here. And so when you have what you see in the black line, that is basically delta F over F over entire population of cells. And here you have this entropy value. And you will understand why we're using this entropy value in a second. Now with the entropy value, we can say and ask, okay, give me the strongest cell here and ask where are the other cells located. And what you see here, what you expect for a visual stimulus, is that they're very closely, if you sort the strongest cell, let's say you sort these guys here, and you ask how far away they are, they're close together, okay? You have this dip. It tells you that something about the structure of this intrinsic activity is not spread anywhere, but it's localized. That's basically trivial for visual responses. Let's see in single trials. In single trials, you see something like that. Over here, you will have at some point a visual response. In this case, you have a non-response over the average, but you have a lot of those dips that happen. And so we can start aligning those dips the same way. In each moment in time, tell me where was the strongest active cell, and so on and so forth. When we do that, we can start seeing, okay, what happens at this moment in time, where you have a dip in let's say, this metric, this population entropy metric, and tell me, this is the spatial extent, and then you have kind of it looks like a visual stimulus, but it's an intrinsic response at this moment in time. And if you ask how the distribution, you see they're localized. And in this moment in time here, where there is no dip, you ask, organize the activity patterns, and you basically have something of salt and pepper. And basically, when entropy is high, there is basically, you don't see much. If entropy is low, it's a way of detecting this intrinsic activity that I was telling you. So what happens? Here, if you look at the population entropy, this metric, and how the mean activity, how strong the cells are, you see a very strong correlation, which means that this intrinsic activity actually, when it happens, it's when there is a strong activity in the patterns. And it seems to be that in some cases, this intrinsic activity is stronger than visual evoked responses that are normally strong. Like small dots will activate the superior colliculus very strongly, but these are, in some cases, even stronger. So basically, the animal is hallucinating. And if you look over here, the mean distance between the strongest 50 cells is lower when the entropy is low. It's saying that there is something depicting about this kind of local activity, like a hotspot that's moving around. This has nothing, this is just a control. All this activity have nothing to do with arousal, locomotion, saccades, and so on and so forth. I don't have time to go through all the controls, but please believe me that we, to the best of our abilities, we haven't found any correlation. And so what can we do now? Since we know how to detect those little events, those intrinsic events that happen sometimes randomly in let's say, in the brain of the mouse. And we can say, oh, sorry, let me go back. We can say, okay, in each instance of time where you have a dip, we can use it as, let's say, a stimulus-triggered event. Normally say like a flash and give me what we've seen before. This is how the brain responds. Now we want to say, when you see this intrinsic activity, how does the brain respond? And what you see is that when you take the average and you ask, okay, which is the strongest cell in the brain, and give me the average, what you see is actually a spatial distribution. This is now rasterized. Each dot is basically, each square there, it's basically, there are a few cells that we put together just for purposes of visualization. But what you see is that you have this strong, basically, cluster event, kind of like a hotspot. If you think of the stimulus onset, and this is for naive animals, you see something very similar, meaning that before the spontaneous, before the stimulus, the activity of the stimulus in a naive animal will mimic the activity of this intrinsic activity. And now this would be like this classical response when you say, okay, this is when the stimulus appear, give me the average over those cells, not where the strongest active cell is, but this is basically a classical response property, spatial response properties. And they are actually similar in size. Now we can do that for spontaneous across, so this intrinsic activity exists in all instances. It's just much more prominent in expert. And if you think of before, this is, so basically this is before the stimulus, it's very similar. At stimulus onset, what you see when you do this analysis of which is the strongest possible cell, you see a huge increase in response properties to the stimulus when the animal is aware of what's happening, when the animal is paying attention to what is happening in the surroundings. So this very much gave us some kind of a feeling of the superior colliculus have been important in saliency, superior colliculus has been important in defining how to move your eyes and connect your gaze. And so we thought, like, okay, this feels a little bit as kind of like a saliency map that is actively moving across, and we're seeing those hotspots, and we wanted to try to test that. And is it something like that, or is it not? And so one way we started doing it is to analyze hit and miss rates. So sometimes the animal licks, sometimes the animal doesn't lick. A little bit of the optical illusion I was showing you before. Sometimes you see the dot that is there, and sometimes you ignore it. And so the question is, let's say, okay, what are the differences when you start looking at this? Let's take a look at the visual responses of this. And so this is the average visual responses of several hit and several miss trials to the same animal. And what you see, this is not just for you to know, it has nothing to do with the reward. We have done the controls. But what you see when the animal has a hit response, basically, you have this prolonged activity over here compared to when you have a miss response, which looks a little bit more like this naive example. There is something on top of the visual responses that is correlated with the animal perceiving the environment. And this is true. This is, let's say, one example animal. This is the hit trials. These are the miss trials. These are all the visual responsive cells. You see that in this animal, most of them are modulated. And this is for across different animals that we've done. So it is a very consistent response property that attention and state modulates how you see the world. And so then Florian was saying, okay, before we're using bigger squares, let's make the squares much smaller to start to see if we can actually depict the difference between this intrinsic activity happening with the visual stimulus. And so now we have very small squares, and the animal has to detect them. It's much more a challenging task for the animal. And if we take the mean of it, you see basically no difference. You see that is the position where the stimulus was evoked. But if you start looking at single trials now, this is the position, this would be miss, this would be hit. And I don't know if you start looking at this, this is very reproducible where the stimulus happened. But when you look at the hit trials here, you always have those bumps of activity surrounding it, which are related with the animal actually initiating the lick response. So to perceive the world, it seems to be that the animal has to somehow be in a state that drives the response, but has this intrinsic, that what seems to be noise, to drive the behavior. So is this a spotlight or is it not a spotlight? And unfortunately, we did some analysis, and it's not a spotlight. It's not that the attention is focused in a particular area of the brain. I don't have time to tell you how we did this analysis, but what we did, we can see where those particular positions are in space in each moment in time. And so we started analyzing if it is something of attention, focus of attention, you would expect before the stimulus in hit trials that there basically these blobs will be looking at where the stimulus will appear. And that's not what's happening. Basically, in the state of attention, they are everywhere, all the time, okay? At least in one hemisphere. I think it might be something of contralateral hemispheric rivalry that is there, but if you look at one hemisphere, there is nothing that tells you that, okay, you have to look over here then to look over there. It's a stage change. It's kind of like you're driving it up. And so what I believe more it's happening, that when you're paying attention, when you're actually knowing that vision is something relevant for you to interact with the environment, you have kind of this prism spotlight. You see, like, there's something will happen, something would happen. And then there's this effect in physics that I saw recently that I think it's basically depicting it. You are heating the system up such that when a stimulus appears, you boom, drive it very strongly. And in physics, there is one phenomenon called superheating. You can boil water, distilled water, and it is in a metastable state, such that whenever you make a perturbation, water is boiling, but it's not boiling. It's hot enough to boil, but it's still not boiling. And then you drive something in, and it starts basically having this superheating effect. And I think that is what's happening right now in the early visual system. This is the synapse just behind the information that comes from the retina. When the animal's being attentive in a particular, when the animal knows vision is relevant, it drives the system up in a state that little perturbation will drive it very strongly, and those strong perturbations will be necessary for the animal to act. And don't try this at home. you can be injured. And with that, I just want to summarize. This is a wide field recording of the activity that I was seeing you before, just for, this is the superior colliculus, and you see this activity happening when the animal is paying attention. And so when you think of how we see, it's important to look at the moment-by-moment dynamics. Sometimes noise has a particular strong relevance, and I don't want to call it noise, but it's some dynamics that are necessary for the animals to be able to interact with the environment. And I don't know if this is the best way of describing it. I just saw this experiment, and it felt that it's depicting some of the activity patterns that we see. And with that said, thank you for your attention.
Thank you very much, Max. It was an awesome talk. Unfortunately, for time matter, we have to close it here.
Oh, one question.
Do you think? Oh,
Yes.
okay, no, the main chairs. So one quick question. Thank you, Norma.
Yeah, I think it was really nice that you showed that it's really not a spotlight, because I was first thinking, like, do you think there's strong plasticity still in the colliculus that could, like, really make these positions extra salient? But then I guess another question would be, do you think acetylcholine could be a good candidate for like elevating the states of all these neurons to like start looking all over the visual field?
So I think it's an intrinsic phenomenon, and we have models for depicting that, and that is driven by neuromodulation. So it could be acetylcholine, I think more of noradrenaline, that it basically changes excitability of the network in a particular way, such that give rise to this dynamics of superexcitability, or like whatever, this globular activity. But I think it's something that happens internally, that you can like push to be active by, and there's if you think of the model that we were describing that I haven't described here, is some sort of surround inhibition. If you change the weight of it, you will see emerging patterns like that. It basically gives you this kind of like activity patterns that you see in a particular state. Acetylcholine is probably also part of it, as we know from the literature in birds, but I don't know the exact mechanisms. My guess is that there's a mixture of different neuromodulators that allow you to come to that particular state.
Thank you.
Thank you.
So this brings us to the end of the session. Thank you to the speakers, to the audience, for the strongest who woke up very early this morning. And thank you all.
Forward. This should be the zoom the pointer. Maybe it has to be on a slide. Okay. They have to. I think they have to click underneath, right?
At the beginning, there is this page is green. So I don't understand why there is no. No, perché prima deve comparire tutto. Questa ora poi la fanno partire. La fanno partire da dietro? Ok. E questo è il pensatore. Sì, sì, e poi uno schiaccia sopra. Okay. So you have to click on your name and the presentation will start.
And then forward, and then.
Forward and e I tecnici lo cliccano. Ah, lo fanno partire loro? Ok. They will.
You move your name over. It's bright. Oh God. It's so bright.
Yeah. So maybe we are. Oh, ciao. Yeah, yeah. Do you think we should start? I know that the room is basically empty, but we are already out of schedule. Okay, so I guess we should start with the next session. So good morning, everyone, and thank you for joining us today's symposium about advancing therapeutic strategies on neurodevelopmental disorders. I am Laura Baroncelli from the Neuroscience Institute of CNR, and I will co-chair this session with Professor Ben Philpott from the University of North Carolina. But without stealing any more time to science, we would like to move to the first speaker.
Yeah. So I'd like to introduce Ludovica Jovino, who is a postdoctoral fellow from the Institute of Neuroscience here at Pisa, and she will be talking about her work to develop a gene therapy for creatine transporter deficiency.
Okay, so good morning, everybody. Thanks for coming. Thanks, Ben, for this kind introduction. Let's say the idea today is to chat with you about one of the main projects in which my lab here in Pisa is involved, concerning a novel gene therapy approach for curing creatine transporter deficiency. And I would like to start with a very brief introduction about the pathology, just for people that are not so familiar with it. So we are talking about a genetic metabolic condition that is an X-linked disorder, okay? The disorder is caused by different kind of loss of function mutation, mainly affecting just one gene that is named solute carrier 6A8 gene, encoding for a very key protein that is creatine transporter. And probably you know that creatine is a quite essential molecule that is actually brought into action in many processes, but the first one is energy production. And this is something crucial for many tissues, especially for those that have like high energy demand, like the brain, for example. For this reason, the main neuropathological characteristic is the complete reduction of creatine, not only at the level of the brain, but especially. And it manifests like many neurodevelopmental disorders with a plethora of symptoms that are quite typical, like intellectual disabilities, autistic-like stereotype behavior, movement dysfunction, and epilepsy. And I would like to stress a concept. So despite many efforts in the last years, we have no cure for the pathology, just some, let's say, palliative approach for managing some symptoms like epilepsy. But you know that to test the preclinical efficacy of a therapy, you need a murine model. And this was exactly what we did like 10 years ago in my lab in collaboration with Cornelius Gross at the MBL. So we produced the knockout model thanks to the ubiquitary deletion of 3 exons, the exons 5, 6, and 7 of the CRT encoding gene. And this actually promotes a reduction in both the expression and functionality of the transporter. And we spent the last 10 years, let's say, in extensively characterizing the model. And what we found is summarized here in this heat map. So essentially, we found a strong reduction in creatine level, not only at the level of the brain, but also in many peripheral tissues. That is the typical hallmark of the pathology. A strong reduction in the body weight, that is something that you can find also in patients, and probably you can appreciate also in the picture on the left. Cognitive defect, autistic-like stereotype behavior, motor dysfunctions, and seizures. And very, very recently, a couple of months ago, I could say, just 1 month ago, I think, we published this very recent paper on brain in collaboration with a group of Alessandro Gozzi, and we found a strong reduction in the connectivity in the knockout model. This is just to stress one critical concept. So we have on the table now a valid model in which we can test the efficacy of a gene therapy approach. But why we believe that gene therapy is promising for curing CTD? And we think that we have at least 3 different reasons. So the first one is well simple. So CTD is a monogenic condition. So we know which is the target to be replaced. The second reason is coming from literature, because we know that creatine supplementation is effective in ameliorating the clinical symptomatology of the other 2 creatine-related syndromes that are the AGAT and the GAMT1, that are mostly related to biosynthesis instead of creatine uptake. And the third one is already coming from literature, because we know that females that are heterozygous for CRT deficiency show, let's say, a moderate to intense phenotype, meaning that there is a sort of dose-dependent relationship between CRT expression level and the intensity of the phenotype. So at the beginning of the story, this is a very, let's say, huge project in which Pisa was the main unit. We decided to inject in the ventriculi of wild-type and knockout animals a new viral vector, so is a AAV9, containing this information, let's say. So what we did was to clone this new plasmid in which we put the coding sequence for the human-created transporter under the control of a small ubiquitary promoter that was the JET one. And I have to state that we have a very great technical limitation, so there are no valid antibody able to recognize the endogenous protein. And to bypass, at least for the, to track, to map the transgene expression, we put in the sequence an HA tag. And what we did was to longitudinally screen this murine model, so wild type and knockout, using PBS as a control, using a plethora of different biochemical, molecular, and behavioral readouts. So as a starting point of the story, so here I reported just for simplicity the data on the knockout brain, but we mapped the biodistribution of the viral vector. And as you can see here, after 40 days from the injection, we stain the brain thanks to the HA tag staining, and we appreciate, a very good biodistribution of the vector along the entire anteroposterior axis, not only at the level of the cortex, but also at the level of the hippocampus and the striatum. But what about the cellular identity? So what about the possible cellular population that express the transgene? And to answer this question, we perform many colocalization analyses. These are like qualitative data, but it's pretty much clear that the transporter that you can see here in green is like this scattered pattern, so like point form pattern that is quite typical for plasma membrane proteins or receptors and transporters that overlap with the MAP2 marker that is typical for neurons, and also, sorry, I don't know what's happening, okay, with the BCAS1 marker that is a marker for oligodendrocytes, and also with microglial cells that we stain with the IBA1 marker. Quite surprisingly for me, when we look at the astrocytes, they actually do not express the transgene. So we decide to go, let's say, furthermore in detail, longitudinally, and to study whether the strategy was actually efficient in ameliorating, let's say, all the endopathological phenotype that I described before. And we started from body weight, that is quite simple. And what we found is that in both the considered time points, so P40 and the later ones, so P140, the strategy was able to ameliorate this phenotype. But more importantly, since I mentioned before that brain created level are strongly reduced, we check whether the transporter is functioning, and we considered 4 different time points, so P20, P40, P100, and P140, and we found a stable recovery of the activity over time, meaning again that the transporter is there and is working. And also when we look at the functional connectivity that, as I mentioned before, this is a part of the work done in Rovereto, at least in the first time point, so at P40, this strategy seems to completely recover the functional connectivity. However, this effect was not so consistent in the later time point. So at the end of this part of the story, we decided to study the more complex behavior. And as I mentioned before, one of the traits of the pathology is autism. So we study autistic-like stereotype behavior, making use of self-grooming. That is one of the most commonly used tests. And what we found was that in this case, the strategy was completely successful in recovering the deficit. And in the same animal, if you look at the correlation between the self-grooming performance and the creatine level, we found a very good correlation. But unfortunately, when we decide to analyze the cognitive performance, we use essentially 2 different behavioral tasks. So the first one was the object recognition test to study declarative memory. In this case, you can appreciate just a, let's say, significant increase, at least in the late time point. But unfortunately, when we look at the working memory making use of YMAs, we were not able to see any type of rescue, meaning that the strategy is like partially successful. And let's say, just before starting with the second part of the story, I would like to stress this result. So if you look at the cognitive performance of the wild-type animals treated with the viral vector, you can clearly appreciate a strong impairment in cognitive performance. Trying to explain this, let's say, possible effect, we reasoned about two different hypotheses. So the first one is that the ectopic expression of CRT, let's say ectopic actually means that cellular population that normally not express physiologically the transporter start to do that. And this could be one possible explanation about this possible toxicity. And the other one is that it's reported in literature that too much creatine transporter could be itself toxic because of an osmolytic action. So at the end of this first part of the story, of the jet story, we are pretty much satisfied because actually demonstrate that gene therapy could be promising for curing CTT, but we have to change our strategy, especially having in mind two different concepts. So the first one is that we have to reach a more physiological CRT expression into the brain, but also to pay attention at the efficacy, but also the biosafety, because we have to avoid toxicity. And in the last 2 years, thanks to a very great collaboration that we built up with the University of Edinburgh, we have the opportunity to develop two alternative plasmids. So the first one allow us to, let's say, answer this question. So we have to reach a more physiological CRT expression. So what we essentially do, cloning this new plasmid that is named here FF351, was to put together many endogenous regulatory sequences. And this allowed to restore the expression in the brain just in those cellular populations that need physiologically to do this job. And trying to pay more attention at the biosafety, so at the overexpression, in collaboration with Dr. Hector, that is here today, so I don't want to spoiler something more about this synthetic regulatory system, but we use, again, ubiquitary promoter, so we are targeting all the cellular population, but thanks to these synthetic regulatory sequences, we are able to pull down the overall amount of the CRT at the single cellular level. And very, very quickly, these are really preliminary data. We started introducing primary cells from the brain of wild type and knockout animals. In this way, we can take advantage of the genotype of our animal. We start from cortical astrocytes, just since they are very, very easy to produce, to transduce, and so on, but we are producing the other cellular population. And what we did in this very simple experiment was to transfect cells using three different plasmids. So essentially the JET1 as a reference point for the first part of the story, and we studied the capability of the new plasmid to deregulate the overall amount of CRT. And what we measured was the intensity, fluorescent intensity, that is like a measure of the expression of the protein in these cells in the three different conditions. And as you can see here, the full endogenous, let's say, plasmid was able to reduce the overall amount of the protein compared to the jet one, while the synthetic one shared like similarities with the endogenous one. As a readout, we measured creatine intracellular level in these cells under basal conditions, so they are untransfected cells, unstimulated or stimulated with CR, so with creatine. And as you can see here, just wild-type astrocytes respond to creatine. And this is an important point because the literature about the physiological expression of CRT on astrocytes is pretty much controversial. But here, the fact that in untransfected condition, just the wild-type cells do this job actually provide an answer. So this is quite promising. But in the second step, we transfected cells with the different plasmids, and then we study the intracellular creatine content in the different conditions. And quite expected, the creatine intracellular level appeared to be strongly reduced compared to the jet one, while if you look at the synthetic, like plasmid, okay, the behavior is quite strange. It's something unexpected. So we are chatting about that, we are discussing about that, but giving the promising results with the endogenous plasmid, we decide to, say, build up a new vector, an adeno-associated vector containing these sequences, and we go in vivo. These are very, very preliminary data, so we are continuing in analyzing data and increase the numerosity. We use Western blot in this case, and after 30 days from the injection, you can appreciate here the, let's say, band with the flag, because at a certain point of the story, we replace the HA tag with the flag one. And after 30 days, the transgene is there. And more importantly, in the same region of the same animals, we found a recovery of the activity of the transporter since the brain creatine level appeared to be, let's say, restored. And when we look at the cognitive performance of these animals treated with a new vector, these are really preliminary. And as you can see here, we have a gap in the group. We are still analyzing the wild type treated with the viral vector. But if you look just at the behavior of the knockout animal treated with the viral vector, you can see that you have like a complete recovery of the wild type PBS animals. And this is true not only for working memory, but also for declarative memory in all the time points that we considered, so the P40 and P100. We're increasing also the behavioral tasks that we commonly use in lab. In this case, we use Morris water maze to study spatial memory, and still very, very preliminary, the knockout with the new viral vector seems to be just like in the middle. So we are continuing the analysis, but if you look at the time spent in the quadrant, which is the platform, they seem to remember well compared to the knockout animals treated with PBS. So as a conclusion, we would like to, let's say, highlight two different points. So the first strategy that seems to be quite promising is pretty much untranslatable. For this reason, we build up a new vector that seems to be very, very promising. And as future direction, we are like consolidating data, starting from the in vitro screening in different cellular population, but also the in vitro experiment will be crucial to disclose potential toxicity. That is something that we plan to do also in vivo, and to complete the behavioral screening. But here the take-home message is yes, gene therapy could be promising for curing creatine transporter deficiency. So I would like to conclude, thanks all the members of my group, in particular my PI, Laura Baroncelli, thanks again for inviting me, and Federica Di Vetta, that is the PhD student working a lot with me in this project, all the collaborators to this project, and thank you all for the attention.
Thank you, Ludovica. We have time for a couple of questions.
Ludo?
Yep.
Thanks,
I'm here.
Ludovica. It was a great presentation and wonderful data. So I was just wondering, with your new strategy, with this new vector, can you say if there is a higher expression in neurons or you saw also microglia cells, and even in which type of neurons? Do you have some specific promoters, or is it just, something?
It's the endogenous one. So I have to check the biodistribution, but essentially this is something that we plan to do in October. And let's say, in this way, this is the only way to check the expression in this case. So what I expect, also because it's reported in literature, that neurons and astrocytes need to express this transporter. So what I expect to find is that, yes, neurons can express, let's say, also based on some recent data in our lab, parvabuminergic cells need to express the transporter, because if you knock out in PV cells, you have a phenotype. So this could be the let's say, the next one, the next experiment. Thank you.
Thanks.
Hi, nice talks. I didn't get from your introduction if males and females are equally affected.
No.
Yeah, if there are also alternative strategies, are you considering alternative strategies to restore the protein function?
Okay, that's a great question. So it's a X-linked disorder, so mainly males are affected. And female, as I mentioned probably in the introduction, as like moderate to intense. It really depends from the kind of mutation and the residual activity of the transporter. So they are not so, let's say, comparable in terms of phenotype, also in patients. And concerning the alternative strategies, is quite, let's say, we try in the past, because you know that, probably you know that cyclocreatine, that is like an lipophilic analog of creatine, is able to cross the the barrier, sorry, the membrane, also without the help of CRT. So this was something that we tried to do in the past, but we stopped this analysis because cyclocreatine is like toxic. Okay, so we can think about other, let's say, cyclocreatine-like molecules, but this is something that we are not planning to do now.
Okay, Ludovica, thank you for the beautiful talk.
Thank
so
you.
just a curiosity, do you have an idea for how long the plasmid can be active within the cells after the injection? Active, I mean it is able to produce the transporters and to be functional.
Okay, I can consider just the last time point in which we performed the analysis, that is P140. So we are talking about 4 months. Is pretty much stable because we measure increase in creatine level and also recovery in the performance. So we have this indication, but the expression of the transgene is quite stable a long time. So this is my indication from the data. Okay, thank you.
Thank you, Ludovica, very much for a nice talk.
Okay, we can move now to the next speaker, which is my co-chair, Ben Philpott, professor at the Cell Biology and Physiology Department of University of North Carolina. He will talk on, well, he will give us new insights on therapeutic strategies for Angelman syndrome.
Great, thank you very much. So I'm really interested in developing a therapeutic for Angelman syndrome, which many people recognize because these individuals have a very happy disposition, but they're also faced with a lot of challenges in life. They usually don't speak a single word their entire lifetime, yet they can live a full lifetime. They have severe intellectual disability. They have severe seizures, they have sleep difficulties, so they need lifelong treatment. There's a therapeutic opportunity because we know that Angelman syndrome is caused by loss of a single gene called UB3A. So if UB3A is deleted, you'll get Angelman syndrome, but it also turns out that if there's overexpression of UB3A, you get another syndrome called Dup15q syndrome, which is a very classic form of autism. So it's very important to regulate your levels of UB3A. It's an incredibly interesting gene because UB3A is only expressed off the pointer, is only expressed off the maternally inherited allele, whereas the paternal allele is epigenetically silenced through this long non-coding antisense. So what that means is that UB3A is expressed broadly in the brain in wild-type mice, but if there's a deletion of just the maternal allele, there's a complete loss of UB3A in the brain. So this presents a therapeutic strategy that we can reinstate UB3A expression by using either a small molecule therapy or CRISPR approach or an antisense oligonucleotide to downregulate the UB3A antisense and thereby turn on the dormant paternal allele of UB3A. To examine and try to identify small molecules that could do this, we took advantage of a UB3A fluorescent reporter mouse. So if a fluorescent protein is fused to UB3A and expressed on the maternal allele, you can look in the brain and the neurons are glowing because that allele is active. If we have the fluorescent reporter on the paternal allele instead, then you see no expression in the brain. So what we did simply was take neurons from these mice that have the fluorescent reporter on the paternal allele, and then we plated them in a 384-well format and added small molecules looking for the identity of an upregulation of paternal UB3A expression. When we did this and screened a small molecule library provided by Pfizer pharmaceutical company, we identified PHA-533-523 as a novel UB3A unsilencer. This had some really interesting properties that made us believe that you can take this forward. First of all, PHA-523 is only working in an enantiomer specific manner. So the S isoform of PHA-533-533 is active, whereas the R isoform is not. And this will allow us to help identify what the molecular targets are of this drug that are causing the unsilencing. What really got us excited about this small molecule is that we can give a single injection at postnatal day 11 and look 2 days later. And in an Angelman syndrome brain, we can see a partial reinstatement of UB3A, not getting back to wild type levels, but a very great improvement over what you see in an Angelman syndrome brain injected with saline. So this is exciting. So it means a peripheral injection of the small molecule compound can produce broad unsilencing of UB3A throughout the brain. And this can just be a single injection that we can give IP, subcutaneously, or even orally that can produce this. When you look in the brain, you can see that if you compare levels of UB3A to wild type, either in hippocampus, cortex, or cerebellum, you see high levels of UB3A expression. You see the loss of UB3A in the Angelman syndrome mice injected with saline. And then once again, if you blow up, you can see the partial recovery we get with this, just a single injection of PHA-533. If you look at the messenger RNA levels, you can see a broad improvement of UB3A levels across the brain. So whether you're looking in the olfactory bulb, the cortex and hippocampus, subcortical regions, including the thalamus or cerebellum, a single treatment of PHA is sufficient to increase messenger RNA levels. We wanted to, of course, show that this has a therapeutic potential for treating Angelman syndrome. So we needed to show that this could work in cells derived from individuals with Angelman syndrome. So to do this, we collaborated with Stormy Chamberlain, who is then at the University of Connecticut and is now at Roche Pharmaceutical Company. Stormy took fibroblasts from individuals with Angelman syndrome, de-differentiated them, and then differentiated them into neurons. When we treated these Angelman syndrome patient-derived neurons with PHA-533, we could increase UB3A levels, and this is acting by downregulating the UB3A antisense that's suppressing the paternal allele of UB3A. So just to give you a summary of this first part of the talk, this shows you that systemic delivery of this new small molecule compound, PHA-533, produces a broad and rapid unsilencing of paternal UB3A in the brains of Angelman syndrome model mice. Importantly, when we give this small molecule to cells derived from individuals with Angelman syndrome, it also produces unsilencing. So this suggests that it truly does have some therapeutic potential. I always give a word of caution because we need to really do a lot more safety and efficacy studies before we could ever bring this compound or a similar compound forward as a clinical candidate. Because currently we don't have enough what we believe is a safety window before when we start to get unsilencing and then when we start to get toxicity. So continued improvements are needed to modify the small molecule to make it a clinical candidate. So I wanted to switch gears for the second half of the talk and let you know that this basic concept of unsilencing the paternal allele of UB3A has really kind of energized the pharmaceutical company who've been working very hard because it's such an ideal target for treating Angelman syndrome. And while we've been working on a small molecule approach to unsilence paternal UB3A, the pharmaceutical companies have been taking forward antisense oligonucleotides, or ASOs, into clinical trials. In fact, there are currently 3 ASOs that are in clinical trials for the treatment of Angelman syndrome. And these ASOs work the same way. Just by downregulating the antisense, they can then turn on the paternal allele of UB3A. So currently, Ionis and now Oak Hill Bio, which has licensed the ASO from Roche, and Ultragenyx are all 3 in phase 3 clinical trials. Now, this is extremely exciting for the community, but there's a problem. It's been very difficult to assess head-to-head which of these ASOs are better and which might in vivo produce better efficacy. And this has been a real challenge for the field. So to address this, what we did is we developed a humanized UB3A antisense mouse to be able to test these ASOs that are in clinical trials head-to-head. And it can also be used for other genetic technologies that are coming forward, such as CRISPR-based approaches or other antisense targeting approaches. So the idea here was we knocked in about 170 kb of the human UB3 antisense into the mouse antisense region. And we wanted to show that first, of course, that this could silence paternal UB3A, just like the endogenous antisense, but also that it could be used to screen the ASOs that are in clinical trial. Really important, these are the 3 regions that are being targeted by the ASOs from Ultragenyx, Ionis, and Roche, and all of them are targeting regions that are in our humanized mouse model. Now, when we did this, we first cultured neurons from our humanized mouse, and we did in vitro dose response curves. And we could immediately see that all the ASOs are not equal. So for example, what we're calling here ASO3 is much more potent than either ASO1 or ASO2, and ASO2 and 3 are much more efficacious than ASO1. So already from our in vitro data, we're beginning to see that there seems to be a clear leader, at least in potency and efficacy, in unsilencing paternal UB3A. Really, what we wanted to do is try to determine how well this could work in vivo. So what we did then is we simply injected the mice at postnatal day 1 with the different ASOs, and we collected tissue at postnatal day 30. And the idea here was that this would allow us to look for the efficacy of the ASOs in vivo. When we did this, we also found something very startling. With one of the ASOs, it was absolutely toxic to the mice. And so when we just looked at survival curves, when we delivered either 5 micrograms or 10 micrograms to the mice, all the mice survived with ASO2, all the mice survived with ASO3, but almost all the mice died with ASO1. And this is a very acute toxicity. And even if we lowered the concentration further to 2.5 micrograms, it still proved to be lethal. Now, I wanted to stress that this doesn't necessarily mean that this ASO1 would be toxic in the patients, because it's always possible that this could be a toxicity that's very specific to mice and might not be found in humans. But at the same time, this was a bit alarming to us and suggested that I would at least move forward very cautiously with using these compounds in vivo. But at the same time, they're already in phase 3 clinical trials, although in one of the clinical trials, they've had to reduce the dose quite substantially from what they originally proposed. Now, what's most exciting, to us at least, is that we can assess in vivo the amount of unsilencing we get of paternal UB3A and the localization and biodistribution of this unsilencing. So just to orient you a little bit, this is UB3A expression in a wild-type mouse, and you can see that it's quite robust. This is a lack of UB3A in an Angelman syndrome model mice, and there's largely a lack of UB3A expression anywhere in the brain, although I would point out that if you zoomed in, you would see that glia still express UB3A because they biologically express UB3A. But what was remarkable to us was what happened when we gave ASO 2 or 3 to these mice. And what we saw is that ASO3 really produced a very broad unsilencing of UB3A throughout the brain. So it was really quite incredible. Pretty much every cell we looked at was expressing UB3A, and it looked very much like what we could see in a wild-type mouse. Now, of course, if you're really an acute observer, you might be able to notice that there are a few differences. So for example, with ASO3, you don't see much staining in the dentate gyrus compared to wild-type mice. And this is probably because a lot of the cells weren't born at the time of delivery because the dentate gyrus is continuously producing new neurons. ASO2 also showed some efficacy, but it wasn't nearly efficacious and didn't produce nearly the broad distribution that ASO3 did when delivered at the same concentration. And it also exhibited some curious laminar specificity, which we would have never have predicted a priori. So if you look, there's pretty strong labeling in layer 4, for example, and some deep layers in 6. So there seems to oddly be some tropism to this ASO, at least in mice that we observed. So to wrap up the second part of the talk, we've developed a humanized UBE3A antisense mouse, which can be used to assess human clinical candidates that genetically target paternal UBE3A unsilencing. So this could be ASOs, it could be a CRISPR-based approach, it could be a zinc finger approach, a base editing approach. Anything that's targeting this area can be studied using our humanized UBE3A antisense mouse. And we think it really presents a large advantage in developing therapeutics because you can use the same clinical candidate in the mice that you'll eventually use in humans. So instead of having to do a target compound that only works in mice and then trying to extrapolate what might happen when you target the human sequence, here you can target the exact human sequence that you'll eventually be using in the clinic. These humanized mice have already proven to be useful because they've revealed large differences in the safety and efficacy of the clinical ASOs. So out of the 3 clinical ASOs that are in trial, we can tell, at least from our mouse studies, that there's going to be a clear winner. And I really think these mice provide a valuable tool to test gene editing and gene targeting approaches, which I think are going to be the future for treating Angelman syndrome. The ASOs are fantastic, and they're really an amazing first wave of treatments, but they also require delivery every 3 months. And these are intrathecal deliveries that are really quite invasive. So the ultimate goal would be to have a one-and-done treatment, such as a gene editing approach. Yet these are also have the disadvantage is that it can't be undone. So we need to test them very carefully in mouse models and, of course, in non-human primates as well. I'd like to end by thanking the people in my lab who are really the ones who are doing this work, and I just take credit for everything they're doing. And then I want to also thank my funding, which includes the Angelman Syndrome Alliance, which is European-based and which is a very active group in Italy, as well as the Angelman Syndrome Foundation and Pfizer and Pinnacle Hill, who supported some of the work with PHA 533. So I thank you for your time and attention, and I see that I've left plenty of time for questions, so I'm happy to entertain your questions.
Thank you, Ben, for the very inspiring talk. There are any questions for the audience? Over there.
Ben, great talk. I don't remember the ASO3 is the one from Pfizer or.
Yeah, that's. I knew I was going to get the question about which ASO is which. So I've decided intentionally not to reveal the companies that are associated with the ASOs at the moment, and the reason is that it could really have a large impact on the financial standings of the company. So I want to wait until it undergoes peer review before we reveal the ASOs to make sure that reviewers don't pick up on anything that they're worried about in the way we've done our studies. But I can tell you that in 1 week, we're going to be submitting the manuscript that will reveal the ASOs. So if it gets accepted quickly, then people will know what the identity of the different ASOs are. And there's also another group that has done a very similar and complementary set of studies, largely using human cell lines and organoids, and they find identical results as us. So we've decided to co-submit the manuscripts back to back. So I think it'll be a very powerful statement because we immediately have replication from another group.
And do you think the function that you see or don't see may be related to RNA structures of the targets? First question. And then the one that is toxic, that is the number 1, yes, correct?
Yeah.
Do you think it's possible that it mess up with with many targets or protein that binds? It binds to RNA, but then it mess up with protein that binds to RNA. Basically, it's messing up with RNA binding proteins, an important one.
Yeah. So to answer your first question, we don't know if it has to do with RNA structure. It's been very difficult to predict what the efficacy of the ASOs are from in silico studies. So it's something that really has to be at least at least in our hands, determined empirically, although I know that some groups are working on getting better algorithms to make predictions. So I don't know if it has to do with RNA structure or really what's causing the better efficacy. I just know that the more ASOs you screen, the more likely you are to have really good outcomes. To answer your second question about the toxicity, we don't exactly know where the toxicity is coming from, but we don't think it's because of an off-target effect. It's really an acute toxicity. When the mice are dying, they're dying within the first couple of hours, really. So it could be potentially one issue is some of the ASOs create very strong polarity, so you need to deliver them with high divalence. So we've started to now deliver this ASO that's toxic with higher divalence, with high calcium magnesium in the solution we deliver it with. And that actually has partially improved the toxicity. So that could be one of the reasons that it's toxic.
Hi. Hi. Thanks for the fantastic talk. So I would like to ask you to speculate or comment on I would say, common problems with gene therapy in neurodevelopmental disorders that it is related to the age of the patient. So I don't want you to spoil the results, of course, of the clinical trials, but what do you think about the timing of the rescue if you have some type of, data, or what do you think about it?
The critical period for intervention is a really important question. So for example, I think the best evidence for this comes from the work of Ipe Gersma, who's reinstated UBE3A at different times during development. And he clearly shows that if you reinstate UBE3A early in life, you can get a nearly full recovery. And if you do it in adulthood, you hardly get any recovery, although I'd point out that you still can get some recovery in certain domains, such as synaptic plasticity, which could be a harbinger for recovery of cognitive functions. So I think you'll get some recovery anytime in life, but without a doubt, the earlier you go, the better. Although I didn't talk about it, we're also working on a gene therapy, just an AAV-based gene therapy for Angelman syndrome. And we see the same thing that Ipe Gersma has seen in his work. When we deliver the gene therapy at postnatal day 1 in the mice, we get very good recovery, albeit still partial. But if we wait just a little while longer, then we hardly see anything at all. So it's going to be really important. So I always think that there's going to be some clinical benefit, no matter when you intervene. The earlier we go, at least in Angelman syndrome, the better it is to be. So I'm looking at the time, and I think I need to cut myself off. And I'm going to go ahead from right here and introduce the next speaker. So the next speaker is Ralph Hector, who is who's a research fellow and coming from the University of Edinburgh. And he's going to be talking about his efforts to develop a very clever gene therapy for Rett syndrome. Thank you.
Thank you again, Ben.
Thank you, Ben. Thanks also to Laura for inviting me to speak. Anything happening? While we're waiting for the screen to come up,
We can start the presentation of Dr. Hector, please.
Yeah, thank you. Yeah, so a number of years ago, we had our first collaboration with Italian colleagues, and our first Zoom call, it was myself, Dr. Paul Ross, and Stuart Cobb. And at the end of the first Zoom call, they said to us that they could understand Stuart perfectly. They couldn't understand me mostly, and they couldn't understand Paul at all. So I'm kind of medium level Scottish, so hopefully we get through this without any problems. And on the first day in the plenary session, Michaela gave a really great introduction to Rett syndrome, which kind of makes this introduction a little bit easier. But just as a reminder, this is a severe neurodevelopmental disorder with a sort of early onset. Children can appear to have normal development up to 6 to 18 months, but then there's a developmental regression and then sort of relative stability. But there's really a lot of very severe phenotypes associated with Rett syndrome. These are lifelong, and there's a pretty severe clinical unmet need. And the cause of Rett syndrome is loss of function mutations in MECP2, methyl CpG binding protein 2. And this is an X-linked disorder, and because of the genetics of this disorder, there's really a very low incidence in boys. It's predominantly females that are affected. There's a mosaic pattern of expression that's X-linked, and so like half the cells express wild-type levels of MECP2 and half the cells don't express any MECP2. And the incidence is around about 1 in 10,000 females. So there are a lot of Rett patients around the world. And as I said, there's a sort of a high unmet clinical need for this disorder. And so, as Ben explained with Angelman, a monogenic, and Ludo explained with CDD, a monogenic disorder does open itself up to genetic therapies if you can either sort of manipulate switch on of that gene, or you can actually deliver it as a gene therapy. And we're a gene therapy lab, and we've been working in Rett syndrome for a number of years. And gene therapy for Rett syndrome, so basically replacing or reconstituting MECP2 expression in these nil cells, is really important to have some therapy. But obviously, if there's no MECP2, you get Rett syndrome. But if you get too high expression, then you get an overexpression toxicity. There's also a really well described and established MECP2 duplication syndrome in humans. So there's a sort of, this is, sometimes you hear this Goldilocks expression. So you have to get the expression of MECP2 sort of just right to be able to achieve therapy, but not move into toxicity. And we're kind of lucky in the field that we've got a big, extensive range of mouse models, some rat models as well. But importantly, there's a lot of Rett phenotypes that are reconstituted in knockout models, so phenotypes that are sort of translatable to Rett syndrome. Also importantly, and this was a lot of this work was done by colleagues in Edinburgh a number of years ago, Adrian Bird and Jackie Guy in Edinburgh. Really importantly for gene therapy, they demonstrated reversibility of Rett phenotypes. So this is a conditional animal model where after a few weeks they're able to switch on MECP2 expression, and then those mice in which MECP2 expression is reconstituted return to sort of wild type still phenotypes. So fundamentally, for somebody who's doing gene therapy, that's a great thing to know. It's good to know that in a genetic model, if you can switch back that gene on, then you can actually potentially recover phenotypes. Some work done again with our lab in collaboration with Adrian and Jackie in Edinburgh. We generated peripheral knockout models, and what this told us was that the brain is the main target. So MECP2 is expressed throughout the body, but if you can target the brain, that's where the biggest, that's the greatest target for the greatest amount of efficacy. So all our studies have been focused on targeting the brain. And then again, just to reiterate, obviously no MECP2 means Rett syndrome, but too much. In every animal study we ever performed, once you get to a certain dose level, a certain level of MECP2 overexpression, you start to get into toxicity. And this obviously has implications for any drugs that you are designing and you want to move forward into the clinic. So in the past few years, we've been working on a genetic mechanism to try and control the level of MECP2 expression in our gene therapy cassettes. And so what we've done is, it's got a couple of sort of classic elements. I mean, the simplest gene therapy molecule would just be a promoter, driving expression of your transgene, and then a poly, and then just to stabilize the transcript. But what we've added into our exact constructs, this is a long acronym, which I think everybody from our lab always struggles to remember that exact acronym when presented with it on the screen. But what we've introduced is two additional elements. So in the intron between the promoter and the transgene, we're expressing a non-mammalian microRNA. Non-mammalian is important so that there's like no off-target effects in the cells. And then in the 3' UTR, we provide complementary recognition sites. So the microRNA is co-expressed by the promoter. So the promoter expresses the full-length MECP2 gene, and it co-expresses the microRNA, and then the microRNA is then able to bind to the recognition sites in the 3' UTR of the construct. And what happens is that you get some transcripts which are then regulated by the microRNA, and this excess transcript is then degraded, and then you get some molecules which escape this regulation, and then protein is made. So sort of a nice theory, but did it work? Well, in the next few slides I'm going to present is all work from this recently published paper. So if you want to download it and look, if you want to ask us any questions, please get in touch with us or grab me in the break. But this is a recently published paper from our group, which sort of describes a lot of the preclinical work that I'm just about to show. So first of all, just proof of concept in vitro and in vivo. We first of all just made some plasmids, and this is MECP2 fused to neon green. And then in the backbone of the same plasmid, we have a second transcription cassette that expresses Ruby. And we have two plasmids here. One of them is regulated by EXACT, our microRNA regulation system, and the other doesn't have any regulation. And so when we put these plasmids into cells and then look at them by flow cytometry, guess what you can see is that in a sort of classic gene therapy system, as the dose goes up, in this case Ruby, then you get transgene expression going up, which is neon green. But in the EXACT regulated cassette, you can see it's much more sort of nuanced and constrained expression. So even as you start going up to these really high levels of plasma transfection, the level of MECP2 is kind of not going up anywhere close to unregulated. And then in the very first proof of concept, where we just sort of, well, there's nothing quick about these studies, but we quickly vectorized an unregulated and a regulated construct and then put these into cells and then looked, and I guess what you can see clearly is that in this tagged version of MECP2, you get really high levels of expression with unregulated, and you get detectable but much more constrained expression with the EXACT regulated molecules. So the question, well, so the proof of concept studies told us that our system was working in terms of regulating, but then the second question was, does this actually confer efficacy and potentially safety? So the first experiment that we did, which was a relatively big experiment, was, and we were just trying to work out what kind of expression levels of MECP2 we were able to achieve and how that would sort of confer efficacy, and also what changes to these genetic elements within the construct might actually make a difference to how strong our expression was and how tightly regulated the circuit was. So we had a series of constructs here, and just to go over this really quickly, we just changed all these elements in blue here were things that we changed the intron, changed the microRNA that was expressed from the scaffold, changed the sequence of the binding sites in the 3' UTR. And in this efficacy study in the knockout animal, we delivered like a whole number of constructs with these different elements. We did this early ICV delivery at P0, a relatively high dose, just to kind of push the system, and even from the start, just say, putting in quite an aggressive dose, like, do we actually get sort of efficacy and relative safety? And in this study, we really just used survival as our sort of main readout. There's obviously a lot of behavioral tests, etc., that you can do, but the knockout animal starts developing these Rett phenotypes quite early, and you can see that the survival is quite short, with a median survival of about 11 weeks. And we just tested all these different constructs. We identified the lead construct that gave us the longest, the biggest amount of survival and efficacy. So this is our lead construct that was selected. So from now on, it's going to be called NGN401, which is, not to put a spoiler, but the name of the clinical product that is in the clinical trial. So this is just in the more details of the NGN401 study. Again, we got this extended survival, really profoundly extended survival, which was really encouraging. There's also a well-established Rett clinical score that labs use to score these mice for different Rett phenotypes. And I guess what you can see here is that compared to the increasing Rett clinical score in the knockout animals, and the green, which is the treated animals, you're getting a score that it's not wild type, but it's much closer to wild type. So you're able to rescue a lot of these phenotypes. And the other encouraging thing was that with these 2 doses, so like in NGN-401, we did 2 doses, relatively high doses, 1e to the 11 and 3 to the 11 viral genomes per mouse. You can see in the vector biodistribution here, and just to emphasize, this is just in the cortex that this data is being shown, that you see a dose-dependent increase in vector biodistribution, but actually you don't see a concomitant increase in protein expression. So it's kind of telling us that in this well-transduced tissue that we're actually exact is working and we're not pushing to higher MCP2 expression levels. And this is just simply a breakdown of this RET clinical score that's broken down into a number of different individual modalities, and then some of these phenotypes, which are recapitulated human. And I guess what you can see is once you start to break these scores down, there are certain scores that are or modalities that are rescued more than others. For example, mobility. So this is all quite encouraging. So that was that's the sort of the efficacy part. In terms of safety, again, just to there are quirks and caveats to the genotypes of these animals, but the most profound RET phenotypes are found in the male knockout mouse, and in the heterozygous female mouse, which is more the genotype of the girls. In patients, there are much more subtle phenotypes and late onset phenotypes. But this is also a good model to test safety in. And what was really striking was that in this study where we delivered 3 different doses of NGN-401, including 1 super high dose, we actually saw really good levels of safety. So in terms of expression, we didn't see increases in expression. Again, this is the cortex, even though the vector biodistribution was clearly going up. And we were able to sort of maintain body weight and have a really low toxicity score in these animals. In contrast, the unregulated construct, which has no exact regulation and is just overexpressing MECP2, we saw really profound toxicity, and at either dose level, these animals died very quickly due to MECP2 overexpression. So we're doing all this work in Edinburgh, partnering with an American gene therapy company called Neurogene, and they were really encouraged by the data and wanted to push this towards clinic. So one of the prerequisites for the FDA is some NHP studies, and this is more difficult in terms of quantification because monkey NHPs and human MECP2 sequence is only 1 amino acid different in NHPs versus humans, so actually quantifying the protein is very difficult. So just even some safety studies, it was in the safety study, NGN-401 was really well tolerated in the NHPs, and we were able to, with mRNA expression this time, just show that, again, purple unregulated, and then green was the NGN-401, that we had sort of achievable regulation in these different brain areas and also in the periphery. So like a good safety profile in the NHP studies as well. So combined, this all came together to give us like a potentially good package to take this forward into the clinic with like little evidence of toxicity, and compared to lots of other gene therapy efforts in MECP2, a pretty good package of efficacy. So one other thing to add, and this is a study that was performed by Neurogene. All the studies that we did that presented so far were ICV delivery in neonatal pups in the mouse models. And our plan was to do intracerebrovascular in the clinic as well. And we know that other companies and other groups also use different delivery methods, for example, intrathecal lumbar. So this is just an NHP study that was performed by the company to give us more assurance that performing ICV was going to give us the biggest potential for efficacy. So I guess what you can see here kind of clearly is that when we look at vector genomes and where the NGN-401 is going to, that we actually get much more. So in the lower lumbar, it's pretty equivalent, but in the upper brain regions where we kind of really want to target, we're getting much greater vector biodistribution with the ICV delivery. So that's one of the main reasons that we're going in with that route. So this is in clinical trial, and so far the dosing has been completed for a number of patients. So 2 patients aged over 11, and then 8 patients aged 4 to 10. And I guess some of the key eligibility criteria for some of these patients going in, and some of the efficacy assessments were provided by the RET clinicians who see the patients, assessments of autonomic function, and some caregiver reports as well. And what's been really encouraging is that there's been improvements in many of the children who've been assessed. So I won't spend too long on this slide just with time, but there's been improvements across multiple scales, improvements in hand function, gross motor function, and communication, which has all been very encouraging, and objective improvements in autonomic domains of sleep and constipation as well. And at this dose, it's been generally well tolerated. One thing just to note is that this is actually data from the end of last year. So at this time point, the patients had all been dosed somewhere between 3 months and a year. And the company is going to put out a press release in the next couple of months, which is going to be the next biggest sort of tranche of data for all these patients. So that will be all these patients with another year of follow-up. So I don't know anything about this data until it comes out, but hopefully it will be sort of a deepening analysis of the efficacy that we sort of saw in this last year. So as I said, unfortunately, this data is a little bit old, but the company are sort of taking their time to put out sort of bigger, more meaningful data sets. So a pretty big update will be coming in the next couple of months. And then this is my last slide. It's just to say that based on the data and based on talks with the FDA, this has been converted to a registrational trial. So this will be hopefully there'll be the proposal is that up to 20 patients will be dosed in the next little while, and there will be sort of more established efficacy endpoints, and that will be the sort of pivotal trial for potential drug approval. So obviously this is a huge amount of work by a lot of people in the team, so I get to present today, but there's been a large number of people in our labs in Edinburgh. Stuart leads our group. He's also the CSO of Neurogene, and then Rachel is the CEO of Neurogene, and we have a great team there who support us, and also some of the clinicians who've been involved with some of the dosing. So thank you very much.
Questions?
Hi, Ralph.
Hi.
Thank you for your beautiful talk. I have a curiosity. Now the Allenbrand Institute released this library of enhancers to direct to have cell-specific transduction with viral vectors. So I was thinking, since the group in Edinburgh proved that you need, MSCP2 you need it basically in the central nervous system, and you don't, all the peripheral stuff are only basically dangerous from the point of view of a systemic gene therapy. So if this could help somehow in the field, I know that now there is a very established vector, but for future development.
Yeah, absolutely. We've done work on endogenous elements and elements that kind of help direct in some more appropriate cells, and there's even things like liver detargeting sequences that you can put on to sort of help this. I guess like at some point with some of these studies, and like I'm showing this data today, but sort of this molecule was locked in like almost several years ago. Like with once you have this molecule locked in, you can't really change it to get these quite long studies of efficacy and safety, and then to get that into trials. So for sure, there is scope to have a follow-up construct that might show even more specificity, more safety, less off target. People are working in capsids as well. So again, that might change the profile of the drug. So I think it's safe to say that there will be another RET molecule that should hopefully be even better than this one. But just in terms of making progress and getting something into the clinic, That's kind of been, that was kind of the goal initially, and yeah, we've got plans to sort of make a hopefully a better construct.
We have time for one more question.
Raf, great talk. So I think I probably missed or misunderstood something. So you're now working with the full-length MECP2 and stepping away from the mini gene.
Yep.
And you're working with the AAVs.
Yes.
And so how did you manage to pack it in because the full length is about 10 kb.
No, so the transcript is 10 kb, but the coding sequence is actually much shorter. It's like 1.5 kb.
Okay, so and why then wasn't used before then? I said, and the mini gene, in your opinion, with the...
So the mini gene was, I mean, initially it was designed just as a sort of concept of like getting it down into like the sort of the main functional parts of MECP2, but that fits into self-complementary AAV. So self-complementary AAV has a much smaller packaging capacity of only about 2.4 kb. So you kind of need to use the mini gene to be able to get that into the self-complementary. Oh, sorry. We use single-stranded AAV, which is the more sort of natural packaging capacity of around about 4.5, 4.8 kb. So actually, when we use the full length, we have plenty of space, if you like, to have our promoter, have our exact circuit, and have the full length MECP2 gene.
Okay, thank you. Thank you. Looks very promising. Thank you.
Well, let's thank Ralph for a really great presentation. Thank you.
And now I have the pleasure to welcome our next speaker, Elena Giusto. Elena is a research fellow at the IRCCS San Camillo Hospital in Venice, and she will talk about pathogenic variants of 14-3 gamma protein in encephalopathies.
Good morning, everybody, and thank you, Laura, for organizing this symposium. As the title suggests, I will discuss how some variants of a protein called 14-3 gamma are involved in the onset of a specific form of developmental and epileptic encephalopathy. So the concept of developmental and epileptic encephalopathies, or DEE, has been introduced for the first time only in 2017 by the International League Against Epilepsy. And it is a result of a continuous and still ongoing process of refinement, which reflect the latest advances made both in clinical practice and the basic research. In fact, before that, DEEs were regarded as epileptic encephalopathy, a condition where the epileptic activity determines severe cognitive and behavioral disorders, leading to developmental slowing and regression. However, since the cause is epilepsy in this case, the treatment with the correct anti-epileptic drugs or surgery can actually revert the clinical outcome of the patient. So, patients with DEEs actually have a component of epileptic encephalopathy. However, they also have a component of developmental encephalopathy, a condition where brain pathology or genetic mutation lead to developmental delay. And in this case, epilepsy is secondary to the underlying pathology. So DE actually represents a combined category where patients present both epileptic encephalopathy and developmental encephalopathy, which are independent to each other. Patients present the first symptoms of DEEs in their infancy or early childhood. And from a clinical point of view, these DEEs are highly heterogeneous. In fact, although the presence of epileptic activity is mandatory by definition, it can differ in many ways. It can differ because of the type of the seizure, because of the anatomical nature of the seizure, the frequency, the onset of presentation, and the pattern of activity. Moreover, the etiology can be different. Although, like we will see in the next slide, most of the DEEs have a genetic component, they may be due to also to structural abnormalities, but also to immunological, metabolic, or infectious issues. Finally, the set of comorbidities that often accompany patients with EEs can actually have a huge range, which can go from mild to severe intellectual disabilities to even autism-like features, but they can also involve some other properties like motor impairment, their behavior. They can have even sleep issues. Altogether, these tell us that we do not have a single disease, but rather a spectrum of disorder. And this makes more difficult both the clinical diagnosis and to have an effective treatment. Because of this heterogeneity, moreover, it is difficult to define a global incidence of DEEs. In fact, it highly depends on the specific syndrome that we are taking into consideration. However, in a recent paper, which is limited to a Wellington area in New Zealand, Poke and the collaborators were able to establish that 169 patients out of 100,000 children below the age of 16 can develop DEEs, which actually means that the incidence is quite high, since 1 out of less than 600 children can develop DEEs. Because of this high incidence and the adverse clinical picture that we can observe on patients, you can understand how this represents a huge burden, not only for the child which is affected, but also for the families who are usually the primary caregivers, and for society as well. And as I mentioned, most of these DEs have a genetic component. And in a recent comparison of epilepsy gene panels, it has been shown how there are more than 900 genes that are related to epilepsy, and 90% of these are actually related to DEEs. Most of these genes encode for proteins that are part of ion channels and that are involved in synaptic function, and most of them are subjected to de novo mutations, which follow an autosomic dominant transmission. Among these genes, there is the YWHAG, which encodes for 14-3 gamma. 14-3 gamma is a protein that is highly expressed in the developing brain, especially in the cortex level, and experimental models knockout for 14-3 gamma have shown how in the absence of this protein, we have reduced brain size and reduced neuronal migration. Up to date, 12 different variants have been ascribed to 14-3 gamma, and all of these lead to a specific form of DE, which is DE56. And in particular, I ask you to remember this mutation, the RC132, because it is responsible for about 20 to 25% of all the cases of DE. Also in this case, this gene is subjected to de novo missense mutations, which act as a dominant negative mechanism, which means that at the end, only 25% of the protein is actually functional. Although I just said that these are actually there is a variable clinical presentation, because although they all have epilepsy and autism-like features, they can differ as for their comorbidities and for the level of the developmental delay. And although I just said that DEs are actually very frequent, DE56 represents an extremely rare condition. In fact, up to date, only 50 cases worldwide have been described for this disease. And I cannot imagine the frustration that a parent can feel when they just know that their child has been affected by such a severe and rare condition. However, one of these parents was able to turn this frustration into action, and he founded the YWHAG Research Foundation. And in this foundation, he not only offers support to the other families who are facing the same issues, but he actually gets together scientists to find the mechanism of the disease and to find new solutions. And in particular, they have already developed some iPS cell lines that we are happy to receive in the next couple of weeks, which carry the the RC mutation, and they are also in the final stages of the production of transgenic mice carrying the very same mutation. But how can a single mutation be so detrimental for the functionality of this protein? To understand this, we need to look a little bit to the structure of 14C3 gamma. So 14C3 proteins mainly exist as dimers, and each of these monomers are formed by 9 helices, which fall to form a cup-shaped pocket, which is called amphipathic groove. And this amphipathic groove is crucial to the functionality of the protein. In fact, although 14C3 proteins do not have their own enzymatic activity, they actually work by establishing hundreds of protein-protein interactions. And this is the reason why they are actually defined as molecular hubs. And what happens when 14C3 gamma interact with a client protein? There can be different outcomes. In fact, 14C3 gamma can stabilize the client protein, which can, for example, increase the enzymatic activity, or they can mask some localization signals. And then in this way, 14C3 gamma can affect the subcellular localization of the interacting protein, or they can prevent further formation of protein-protein interactions, or on the opposite, they can actually work as a bridge to increase the protein-protein interaction. And last, they can also work as chaperone-like proteins, thus assisting the folding and preventing the aggregation of the other proteins. So considering the widespread distribution of 14C3 proteins and the importance of these interactions, we can understand that when there are some circumstances in which these interactions are interrupted, we can witness the onset of several kinds of disorders. And one way to interrupt this interaction can be, for example, phosphorylation. In fact, in our lab, it has been previously shown that when 14C3 gamma is phosphorylated by a kinase which is called PAC6, we can have the dissociation of the monomers and the release of the target protein, in this case LRRK2, which, as you may know, is involved in some familial form of Parkinson's disease. In fact, as you can see in this overlay assay, where LRRK2 is immobilized in a membrane, when it is probed with the unphosphorylated 14C3 gamma, we can see that there is a strong interaction between the two proteins. However, when it is probed with the phosphorylated 14C3 gamma, this interaction is basically lost. Now we are actually checking the effect of phosphorylation of 14C3 gamma on its interaction with alpha-synuclein. And in this case, we saw that although both the unphosphorylated and the phosphomimetic 14C3 gamma can decrease the number of cells that have alpha-synuclein aggregates, the unphosphorylated form has a higher effect on reducing the size of these aggregates, thus suggesting that the phosphorylation can actually affect the chaperone-like properties of 14C3 gamma. We more recently had a look to the global interactome of 14C3 gamma upon phosphorylation. And as you can see, we found some targets, which are indicated in blue, who actually represent those proteins that lose interaction with 14C3 gamma upon phosphorylation. We put together these results of protein arrays and proteomics, and in collaboration with Claudio Manzoni at UCL, we performed some gene set enrichment analysis, which allowed us to identify among these hits that I just showed, these proteins that are known also to be involved in the onset of neurodegenerative disorders. Altogether, this tells us that the perturbation of 14C3 gamma interactome can actually deregulate some pathways and finally lead to the onset of disorders. We therefore wondered if this could actually be also the case for DEE. In other words, is DEE the result of a loss of interaction between 14C3 gamma and some unknown targets? To answer this question, we went back to our proteomic data and found that among the strongest interactors of 14C3 gamma, there is ADAM22. Actually, the interaction between ADAM22 and 1433 proteins has already been reported in the literature. ADAM22 is expressed in the brain at the level of the cerebellum and of the cortex, and it is involved in synapse maturation, transmission, and myelination. Very importantly, mutations at the level of ADAM22 have been related to some forms of epileptic encephalopathies. And how it is work? So basically, when ADAM22 is bound to 14C3 gamma, this is translocated to the membrane of the neurons where it can interact and form heterotritrimers with LGI1. And it has been shown that basically 14C3 gamma works to stabilize the interaction between ADAM22 and LGI1. And when this interaction is somehow lost, in this paper they observed the onset of epileptic seizures. So we asked ourselves if the mutation that I told you earlier could be enough to promote the loss of interaction between 14C3 gamma and ADAM22. We therefore performed some co-immunoprecipitation experiments, and as you can see, when ADAM22 is co-expressed with the wild-type 14C3 gamma, the two proteins can interact, but when it is co-expressed with the mutated form of 14C3 gamma, we lose this interaction. And again, we validated this by means of overlay assay, and we see that, again, with the wild-type 14C3 gamma, we have a higher interaction with ADAM22. So to come closer to the focus of this symposium, we asked how can we translate this into a preclinical setting. And we think that one way could be the use of small molecules. In fact, small molecules can work in two ways. They can work as protein-protein inhibitors, preventing interaction between proteins, or they can work as molecular glue molecules, which means that they do stabilize the interaction between interacting proteins. In this context, we have just started a collaboration with Professor Christian Ottmann, who happens to be not only a great expert in the biology of 14C3 proteins, but also the co-founder of the Amagon Therapeutics, which is a company focused on the design of glue molecules for 14C3 proteins. In fact, thanks to this collaboration, we were able to have some very preliminary results where Dr. Ottmann was able to screen a library of 9,000 molecules, and those that you see in green are actually some candidate targets that are able to stabilize the interaction between 14C3 gamma wild type and the peptide that represents ADAM22. Of course, we need to validate this result by using the modified form of 14C3 gamma. So, in conclusions, we saw that DEEs represent a highly heterogeneous category of epileptic encephalopathies. We saw that DEEs 56 is one of these form of DEEs, and they are due to some variants of the 14C3 protein. And in particular, the 14C3 gamma with the RC132 mutation is the one responsible for the majority of the cases, and it is related to the interaction with ADAM22. And we hope that in the future we will be able to reverse the interaction between the mutated form of 14C3 gamma with ADAM22. I would like to conclude by thanking my PI, Laura Civiero, and lab members, the sponsor, and you for your attention.
Questions from the audience? I have a curiosity, actually, Elena. I was wondering if there is information about the developmental profile of expression of 14C3 gamma, and if this expression has some feature of cell specificity.
So they are mainly expressed in the developing brain, and the expression declines after the early stages.
And about cell specificity of the expression?
No, they are ubiquitously expressed. So there are actually several isoforms of 14-city proteins, and they may have some redundancy, but they have also some functional specificity. And in this case, it is specific for the gamma isoform.
Thank you.
Thank you.
More questions? If not, we can thank one more time Elena. And this brings to the end of this session. I would like to thank one more time all the speakers for their wonderful talks, you in the audience for the productive discussion, Ben for being my co-chair, and well, enjoy the rest of this conference.
Hello. Yeah. Sorry, I should not have closed it. I didn't want to. Ah, stai vedendo tu?
Aveva abbassato.
Ah. Sorry.
Perché se no non esce.
Your name.
Non c'è?
Do you have a pointer or do I use the mouse? Use, HDMI. Do you have an adapter?
The presenter is.
Yeah,
Bruno, lo vedi? Ah?
Buongiorno.
It's okay?
Yeah, it looks fine.
Okay.
Looks fine. Are you taking care of flights? No, not you. Okay.
You want a glass?
No, it's my bottle.
You have your bottle? Ok. Quindi questa gliela
What is
togliamo.
this? What is
Questi
that?
sono cose. Yeah. Okay.
Ora?
Can I ask you something? After this, we will have your words.
Yeah.
Can you stay because the young people will be very pleased if you stay
Sure,
on
of course,
stage?
of course.
Okay, if you don't mind, no?
Of course not.
Okay. So I need to have the people in, so and then we start.
I still don't have my presentation.
Ah, bah, quindi siamo a posto. Ah, mais alors, c'est parfait, hein? Oh, they are all on.
Questa è presentazione, ma noi non la vediamo. Ma butti fuori, Blu. Vediamo questo.
Okay, so maybe we should
They're all in
restart.
the little party. Look.
Yeah.
It's ridiculous.
Ok. Ok.
Good.
Presente?
Yeah. What's that? What is this?
It's laser point.
What does it work?
Non agisce sul sole.
It doesn't work.
Sul sole non va.
Should be remote. Yeah, it is there.
Però ora sì, non lo vede con il sensore.
Pointer.
Devo mettere la macchina rossa. Eccolo, puntino piccolino, vedi? Un puntino piccolino.
Wow. Yeah, the color is the. I don't know. Pointer. So. I don't know if you can change them. Laser. Maybe it's got better.
Sempre piccolo.
Kind of. Okay. Good. Everything seems okay. Good.
It is? Daniel? Okay. So we let them in, okay? One more minute and we start, so. Okay. So, wow. We came to the end. It's incredible. After all these days. So it's really a honor for me to sit in this chair again at the end of our Congress. And let me say that it's been an honor for me that Daniel Choquet accepted to deliver the presidential lecture. Daniel Choquet, as you all know, is a pioneering French neuroscientist whose work has transformed, really, our understanding of synaptic organization, synaptic structure, function, and plasticity. Daniel grown up in Paris, and actually grown up in a family, if I may say, of outstanding scientists. His father was a brilliant mathematician, and his mother was a physicist, and she was the first woman ever elected at the French Academy of Science. So let us say that when you're grown up, outstanding science was sort of bread and butter in the family. Daniel Choquet has been trained as an engineer at École Centrale Paris before obtaining a PhD in neuroscience at the Pasteur Institute. And then he joined CNRS in 1988, and afterwards he did a postdoc at the Duke University with Michael Schitz, studying how mechanical forces shapes cellular structures. And then he returned to France to establish his own research group in Bordeaux, focusing on high-resolution imaging of transmitter receptor trafficking. Daniel's most influential discoveries revealed that neurotransmitter receptors, and particularly AMPA receptors, are not fixed in place, but move dynamically within neuronal membranes, entering and exiting the synapse in a process that is critical for learning and memory. And with these discoveries, it gave us an unprecedented insight into nanoscale organization of the synapse. Let me say that behind this research, Choquet has played a transformative role in building neuroscience infrastructure and research areas in France and Europe. He founded the Bordeaux Imaging Center and the Interdisciplinary Institute of Neuroscience, again in Bordeaux, creating platforms that support cutting-edge research in neurobiology. He has an incredible CV. It's too long to say. His work has been widely recognized with numerous honors. Let me say a few. He has been awarded by the CNRS bronze and silver medals in different times. He is an awardee of VRC Advanced Research Grants for 3 times in a row. He has been elected to the French Academy of Science, in EMBO, in Academia Europea, and has been awarded of the Grand Prix of the Académie Nationale de Sciences, Belles Lettres et Arts de Bordeaux. And he was also named Chevalier of the Légion d'Honneur, and in 2021, he was named Officer of the Ordre of the Des Palmes Académiques. Let me say that Daniel Choquet is not only a visionary scientist and an innovator who has really reshaped our understanding of synaptic mechanism, but really I want to say that he is also a generous scientist, a trust colleagues, and a real friend. Thank you, Daniel, for being with us today. Daniel.
Yeah. Thank you very much. Thank you very much for staying to the very end of the meeting. I'm sorry again, I was supposed to speak the first day. I couldn't make it for personal reasons, but I'm really happy to see that there are numerous people still there. Monica, thank you so much, first for your kind words today, for inviting me here. It's really an honor to deliver this presidential lecture. And thanks also for your unconditional support over those last more than 20 years. You have to know that Monica has been the most active supporter of the Bordeaux endeavor when we just started more than 20 years ago. And so hopefully you're not disappointed with what we've been able to do. Actually, Monica invited me many years ago to a presidential lecture, or to a lecture. I don't know if it was presidential, but an important lecture at the Fence. And that was nice. And so hopefully, although the topic is similar today, the data is going to be different. We were discussing with one of your colleagues in Milan today, Eduardo, that indeed I've been working on this field of AMPA receptors for now about 25 years. So of course it's a lot for a single receptor, but what I'm going to try to tell you today is that the complexity of these receptors, their composition and how they are organized and how they participate to the regulation of synaptic function and synaptic plasticity is more complex than ever. And actually, we really have to realize that we have only scratched the surface of the role of these receptors. And I won't present that today. I had to make choices, but also it's important to know that there are more and more diseases which are linked to these receptors and the way they traffic. Actually, I was this morning in a very interesting session on neurodevelopmental disorders, and there are many, many developmental disorders, autism linked or mental retardation linked, which actually directly involve AMPA receptor trafficking. And hopefully in the coming years, I will be able to have a Polish stories to tell you about. So today it's really going to be about the basal, the basic, I would say, function of these receptors. And but most of the talk is going to be unpublished data. So hopefully you will enjoy it. So let me start. These timers, I tend to go over, so I'll try not to. So first, a very brief introduction, but certainly for this audience, it's not really necessary. Let's see. Yeah, I'm trying to, no, it doesn't work. Trying to get the pointer to work. No, it doesn't really work. So I'll do it by hand. Yeah, there's no really need to introduce that. Just a few details. You all know that AMPA receptors are really the workhorse of excitatory synaptic transmission, together with their regulator-associated partners, NMDA receptors, kinase receptors, and metabotropic receptors. They are well known. I'm sure most of you know all these properties. They are cationic channels. They are either homotetramers or heterotetramers. Their permeability to calcium can be different depending on their composition, although recent work seems to suggest that all AMPA receptors are permeable to calcium to some extent. They come from 4 genes, GLUA1 to GLUA4, and there's really, as you'll see in a second, a wide variety of assemblies of these subunits that forms these tetramers. We have a pretty good idea of their structure, thanks to the pioneering work of Eric Guo, more than 10 years ago now, showing that you can really separate those receptors in 4 parts. The amino-terminal domain, that is still a bit mysterious in terms of its function, but I will tell you towards the end of the talk some suggestions for the probably extremely important role of this amino terminus. Then the ligand binding site, where all the 4-glutamate bind. And what's very important, and I really want to stress that, is that first, the affinity of those receptors for glutamate is pretty low. It really depends on the composition of the receptors, but for a typical GLUA1, GLUA2 receptor in the hippocampus, for example, the affinity is in the order of a few hundred micromolar. And the other factor that's very important is that you need at least 2 glutamate bound to open receptors, and the more glutamate are bound, up to 4, the more open becomes the channel. And this is also important, you'll see in a second. Then the transmembrane domain, and an unstructured C-terminus that's very important to regulate receptor trafficking. AMPA receptors don't come alone as GLUA subunits. They are actually part of a very large complex that's composed of a wealth of auxiliary subunits. This is the cryo-EM structure of AMPA receptors, together with one of the main ones, the TARP gamma-2. You see that TARP gamma-2 is closely associated with the receptors. They regulate their function, their biophysical function, their trafficking, and they are also the target of various diseases. Actually, we don't know that much about the cell biology properties of these auxiliary subunits, because as you see, they are really embedded in the membrane and are barely accessible to any ligands because of the way they are closely packed with the receptors. And that's something else I won't talk about today. Can't talk about everything, but we are really developing ways to be able to label endogenous auxiliary proteins in live neurons. So now if we go to the organization of these AMPA receptors and how they actually regulate synaptic transmission, this is cutting short 20 years of work. We all know now that receptors are reversibly stabilized in the postsynaptic density through a variety of complementary pathways. First of all, of course, their stabilization by intracellular scaffold partners, essentially through auxiliary subunits, and as you'll see a bit later on, also through extracellular interactions, which are not represented here. And then they go in and out of the synapse through a variety of trafficking pathways, of course, internalization, externalization, but also lateral diffusion, and that's been the main contribution of our group 20 years ago. And actually from a number of data, and in particular the fact that the sites of internalization and externalization are largely outside of the postsynaptic density, it's really the synergy between this intracellular transport, exocytosis, and endocytosis outside of the PSD, together with this diffusion trapping and interaction with slots or trapping slots here in the PSD that determine the number of receptors in the postsynaptic density. So this determines the efficacy of synaptic transmission together with the probability of release of transmitters. And as I was telling you earlier, because the affinity of the receptors for glutamate is pretty low, the area over which receptors get activated is pretty small. And that's something that really people oversaw for many, many years, but that's extremely important in terms of the function of the receptor. And it really has to do with understanding the dimensions of the PSD, the dimension of glutamate diffusion, and the affinity of the receptors. So nobody's ever been able to actually measure the area over which receptors are activated. We know since decades that there's about 10, 20 receptors which are activated upon release of a vesicle. And for a very long time, people have thought that this was relating to the number of receptors present in the PSD, and that actually the PSD might be saturated by glutamate. And actually work from dozens of labs, essentially modeling work, but also some physiology, really show that it's only a fraction of the receptors that are activated by glutamate because of this low affinity and cooperativity. And so this is a simulation we did some years ago, and many people have seen the same thing, that the area over which receptors are activated, the area over which there's enough glutamate for receptors to be activated, is about less than 100 nanometer full width maximum. And knowing that an average PSD is in the order of 300, 400 nanometer, that means that upon release of a single vesicle, only about 10% of the PSD sees enough glutamate to be for receptors to be activated. So of course, there are cases of multivesicular release that could occur at the same terminal. But as we know, in normal conditions, most synapses, at least in the hippocampus, in the CA1 region, our favorite region of study, the Schaeffer collateral to CA1 pathway, it's mostly single vesicular release. And so this conundrum was actually kind of solved some years ago, about 10 years ago now, with the discovery, and this is only a scheme for now, I will present you some data a bit later on, with this idea that I'm sure you've heard about because it made a lot of noise in the field, this idea that actually there might be a correlation between the organization of the postsynaptic receptors and the sites of release. And the first thing I'll show you a bit later on is that indeed receptors are concentrated for a part in small nanodomains. And there's been data showing that these nanodomains might be somewhat related to the sites of release. So this would somewhat favor the activation of receptors in front of those release sites. But as you'll see, the story is probably way more complicated than that. And I will give you some recent elements for that. So the message to retain from this introduction is that if you are a receptor, if you're located in front of the release site, or if you're located just a few tens of nanometer away, it makes a huge difference because in one case you're activated, in the other case you're not activated. And so that really signifies that when you calculate this amplitude of the postsynaptic EPSP, it actually really depends not only on the total number of receptors in the PSD, but also on the location of these receptors at the nanometer scale. And this concept, in my view, which emerged maybe somewhat like 10 years ago, is really a game changer in terms of understanding the regulation of synaptic function, because not only you need to measure transcripts of receptor, but you need to measure the surface protein numbers, but you'd also need to measure where are the receptors if you really want to have a good assessment of the efficacy of synaptic transmission. And if you only remember that from my talk, that will be already a good achievement for me. So I will tell you 3 stories. As I said, there are most of them unpublished or only partly published. I will first tell you about our attempt to actually look at where all the hundreds of different subtypes of receptors are localized. humbly starting by just a few of the subtypes. Then what are their dynamics and our efforts at looking at the receptor dynamics in intact tissue. And then finalize with new data from an old finding that we had that for a long time remained a bit controversial and not so well adopted by the community, that the diffusion of receptors and their diffusion trapping actually contributes very importantly to short-term plasticity. And I will show you our new data and molecular mechanism for that. So first of all, what about intersynapse and interneuronal diversity of AMPA receptor expression at the global level, at the cell level first, and then at the synaptic level, comparing excitatory versus inhibitory neuron. So as you know, there's a big variety of AMPA receptor complexes. I told you this is data from the Allen brain, transcriptomic maps of the whole brain for AMPA receptors here in the top, the 4 GLUA subunits, and then the over a dozen type of proteins which have been shown relatively robustly to interact directly with AMPA receptors and which are called AMPA receptor auxiliary subunits. And so it's very evident from this graph that while the subunits are pretty much expressed in all different subtypes of neurons, at least at the transcriptomic level, there's a pretty large diversity in terms of auxiliary subunits. And so when you look at the combination of all these, you realize that there's not 1, not 2, not 5, but dozens and dozens of different subtypes of AMPA receptors. And that's really a challenge actually for the pharma industry to develop ligands which are going to be able to be specific for each of them and be expressed in specific subregions. So this is at the whole brain level. And actually I discovered myself how to dig out in these maps just a few days ago from new data we are going into. This is the same map, but now concentrating on hippocampal neurons. You see this diversity. And then the same map now focusing on CA1 transcriptome, pyramidal neurons from the CA1. But you see that even in this small region and small subtype of neurons, the pyramidal neurons, the variability is still pretty high. So that's good. We have all the answer we wanted, you may think. And I'm going to tell you that not at all. This is transcriptomic data. It's very interesting, but it might be a bit limited. And one perfect example for that is the expression of GLUA4, or GRIA4 as the gene for GLUA4, that you see is expressed pretty high in some subtypes of excitatory pyramidal neurons. And actually, it's been known for decades, and I'll show you more data about that, that actually those pyramidal neurons do not express on the cell surface this GLUA4 protein for unknown reason. And so just a word of caution on transcriptomic data, that despite its huge power, it does not tell you where is the protein. And that's been our obsession for many, many years, is to really look at the proteins expressed on the cell surface to know where they are expressed and how they are regulated. So for a long time, the field was really plagued by the absence of tools. Early on in the 90s, when the receptors were cloned, the first series of antibodies that were done were all against the C-terminal of these receptors at the time where we didn't really know the topology of the receptors. And it was much easier to make antibodies against the unstructured C-tail of the receptors. And the problem is that these are completely useless in terms of looking at the surface expression of the receptors, because those AMPA receptors, they exist in maybe, depending on the neuron subtype, maybe only 10-20% of the receptors are on the surface. The vast majority of the receptors are in the ER and Golgi. So if you have to stain a preparation with intracellular ligands to your receptors, you have a very poor view of really what are the surface expressed functional receptors. So then there's been a few antibodies to the extracellular domain of the receptors that became available in the year 2000s. They were very poor, but really a gigantic game changer came from the structural biologists, and in particular Eric Guo, but others now, that for their preparations in order to be able to crystallize the domains of the receptors, and now in order to do cryo-EM, purify the receptors and being able to orient the particles at the cryo-EM level, where they had to develop ligands for the endogenous forms of the receptors, the intact structured forms of the receptors. And so they came up little by little with specific ligands for the different subunits with very high affinity. And I've been extremely fortunate to start to collaborate with Eric Guo very early on, more than 15 years ago, discovering these game changer antibodies. And so we have now, thanks to Eric, high affinity ligands for all the subunits of the receptors. And so we are using those to look at where are the receptors at the cell surface and how they are organized. So first, to actually characterize the property of these ligands, we produce them or we get them produced by Eric, whatever. We used amino terminal domains expressed in heterologous cells to verify the selectivity of these ligands. These are a series of COS cells expressing the various ATDs of the receptors and labeled with the various GLUA fabs. You see extremely good selectivity, no cross-reactivity whatsoever. And thanks to the work in collaboration with Jonathan Ellegeert, our neighbor, a structural biologist, we can do SPR to measure the affinities. And really, our Rolls-Royce antibody is this Fab anti-GLUA2 that's now freely available, which has a 1.5 nanomolar affinity as a Fab fragment. So it's really unheard of in the field. And it's extremely useful. And you see the other ligands for GLUA1 or GLUA3 or GLUA4 are a bit lower in affinity, but still good enough to do very good immunocytochemistry. So with these tools at hand, we went and stained our favorite cultures, the cultured hippocampal neurons from mouse or rat. And we were really stunned by the image that we saw, because that's a typical staining from a 2 or 3 week old cultured banker cultured hippocampal neuron. And each of the Fab fragment is labeled with a different color. And so the mixture of these colors give you this rainbow of colors. And you can immediately see that each neuron seems to express a pretty constant ratio of the various subunits, but the ratio of the various subunits in between neurons is extremely variable. You have green neurons here, which seems to express predominantly a GLUA3. You have pink neurons that have a lot of GLUA1. You have these violet neurons that probably express equal numbers of all subunits. So this was extremely striking, and we decided to invest that a bit more in detail. So first, it was very obvious we had at least 2 different main types of neurons. These neurons, which are very green, express a lot of GLUA3, and those neurons that express more equivalent numbers of A1, A2, A3s. We looked at the distribution of the expression of these receptors in between synapses now first, by doing line scans or whole neuron analysis. And this is, each dot here is one synapse over a given neuron. You see two examples, this top neuron here and this bottom neuron here, this green one. And you see that the ratio of these various subunits is extremely constant over all the neurons. So of course, although there are very weak synapses which express very low number of receptors, very strong synapses that express very high number of receptors, the ratio of the different subunits is extremely constant, or relatively constant over the whole neuron. But in between neurons, the ratio is very different. So of course, we wanted to know, is that a general finding, and what are the different subtypes of neurons which are expressing these different ratios of subunits? And so this is really an ongoing study. I'll give you the results that we have for now. Of course, the obvious first candidate to look at was to compare interneurons with pyramidal neurons. And so for that, we use GAT67 as a marker of inhibitory interneurons. We compared hippocampus and cortex with not major differences I would say there are some detailed differences, but not major. So I will stick to hippocampal data from then. And so the study that we've done, that's finalized now, is just to compare GAD positive and GAD negative neurons and compare the staining amplitude of GLUA1, GLUA2, and GLUA3. And because we have all these neuron subtypes in the same dish, we can really compare absolute amplitudes. And the finding, I think, which is kind of interesting, is that in interneurons, the amount of GLUA2 is about relatively the same. And the aficionados in the AMPA receptor field know that interneurons have been known for a very long time to express essentially calcium permeable receptors. And so there was this idea that because calcium permeable AMPA receptors are GLUA2 lacking, then this would mean that indeed there would be no GLUA2 in interneurons. And so we claim that's not true. On the contrary, you have the similar amount of GLUA2 in interneurons and pyramidal neurons, but you have a really large difference in GLUA1 and GLUA3 expression, with interneurons expressing way more GLUA1 and way more GLUA3 than GLUA2, hence making much more GLUA2 lacking receptors. So this still holds true. This is just another representation of that, looking at the ratios of A1, A2s. You see a big difference in ratio between pyramidal neurons and interneurons, relatively similar level of expression of A1 and A3, and same, more A3 than A2. So in summary for this first part, really remember that if you want to be precise and state things about expression of subunits, you really have to go to the protein level and label surface proteins, and the tools are available now for that. The regulation of the subtype of receptors is not done so much by a downregulation of A2, but more by an upregulation of A1 and A3. The last player I mentioned to you is A4. Actually, we got the A4 antibody available only recently because it was very difficult to actually make a specific A4 antibody for Erigo and actually other people. But we do have now a very nice A4 antibody. And what Chloe found recently, using that very nice new GLUA4 antibody, is that indeed it's PV interneurons that express massively GLUA4, something that was known before, with virtually no expression of surface GLUA4 in the other neurons, which are the non-PV interneurons, but also the pyramidal neurons. And so this is really to stress that this is vastly different from the image you would get from just looking at the transcriptomic data. So let me switch now to the second part of this talk, which is the nanoscale dynamic organization of the receptors. And I'm going to go very quickly on the first part because it's all published and I've already discussed the conclusion. Just to remind you that this new view of the nanoscale organization of the receptors started in 2013 when three groups, my group, the group of Tom Lampier, and the group of Fukata, discovered simultaneously that AMPA receptors are not diffusely distributed in the PSD, but are for a large part concentrated in nanodomains. So see, this is a super resolution method that's called STORM, but you find the same thing, whichever method, super resolution method that you use. Actually, most people now use TED, which is good enough to see those nanoclusters. You have around half of the receptors are clustered in those small domains, and the other half is more diffuse in the membrane. We see those clusters also by pre-emitting immunolabeling by EM, even with cryo-EM, some recent papers see those nanoclusters. There's a lot to discover yet on the properties of these nanoclusters. They may be formed secondary to condensate and protein segregation. I don't have time to discuss that today if I want to stay on time, but just to know that they are relatively homogeneous in size. And what's interesting is that if you look at the number of nanodomains per spine, what you see is that as the spine grows during plasticity, in particular during increased potentiation, what you see is an increase in the number of domains rather than a change in domain size. Then the other thing, and that will be my only point on these nanodomains, which is something that big breakthrough that was done by Tom Blampied in 10 years ago, actually, exactly, is the finding that there is a good correlation between the location of the postsynaptic receptors and postsynaptic clusters of PSD95 and the presynaptic release machinery. So that created this concept of transsynaptic nanocolumns. Many groups have been excited by this finding and are working on that now. The group of Mathieu Dalva, using STED, showed that indeed when you do plasticity in a slice, what you do is that you increase this number of nanomodules. We found the same things ourselves, and we were able to actually misalign those receptors by playing with the adhesion proteins. And lastly, what people are doing now is really looking at the various proteins of the PSD to see where they are. It's a long endeavor. We are far from finalized there. Probably the most interesting finding that's been reproduced now by many groups that we published a few years ago is that the main two receptor types of the PSD, glutamate receptor types of the PSD, AMPA receptors and NMDA receptors, are really segregated. And this is actually regulated by neuronal activity. But I don't have time to tell you that. So the story might seem simple, except that more and more people became interested in that, which is good. And in particular, structure people became interested in that and trying to do in situ tomography and in situ structural biology. And there's been a few papers out now that actually modulate a bit this nanocolumn change. So I'll just show you this piece of data from the Branger lab, which is still in bioRxiv, showing that with cryo-EM and being able to label endogenous receptors with knock-in mice and nanogold labeling, they actually found that the location of the duct vesicles is actually not co-aligned with the clusters of receptors. There are clusters of receptors there and there. And what you see here is that actually the receptor clusters are not exactly in front of the vesicles. We don't really know what this means. Not all the vesicles might be actually released. It might be a bit like at ribbon synapses where, as you know, the vesicles are docked a bit away from the receptors and the calcium channels at the nanometer scale. But still, what's been shown in various modeling studies is that even if there is a small bias of the vesicle location with respect to receptor clusters, that's actually enough to favor synaptic transmission. And so my guess is that there's actually going to be a regulation factor there, a new regulation factor whereby the actual localization of the receptors at the nanometer scale with respect to the release site is going to be able to regulate synaptic transmission. And one can forecast, for example, that processes of synaptic potentiation and synaptic depression don't absolutely need to completely remove or increase the number of receptors in the PSD, but just by shuffling receptor organization at the nanoscale, that's enough to change synaptic efficacy by a factor of 2 or 3 at least. So now let me move to the final part, which is divided in two, I would say. First of all you've seen are static images of receptors, which is very important. But as many of you know, what I really like is dynamics. And we've shown from very early on, and Monica, thank you for mentioning that, we've shown that those receptors are extremely dynamic in the plane of the membrane. Actually, AMPA receptors are not the most dynamic receptors. The most dynamic receptor types are more metabotropic receptors, which are not accumulated at the PSD. But AMPA receptors are certainly way more mobile than NMDA receptors, for example. And you can really see them entering and leaving the synapse at pretty high rate for at least part of them. And you'll see our current model. So this was for many years limited to cell cultures. And of course, we've been told over and over that this might be an artifact of cell culture. And so I took that as a challenge, of course. And we've took this endeavor very long time ago now, but we are really getting to a success to really be able to measure mobility of endogenous receptors in brain slices. And that's been more than a 10 years endeavor by many people. And currently, in particular, Angela Getz, who's now a PI in the Netherlands, in Amsterdam, together with Mathieu Ducrot, who is an optical engineer who helped us develop the lattice light sheet, who's actually the technique that allowed us to do that. So although this is published, I really want to emphasize how useful the method is. It's a method to label a subpopulation of neurons in a tissue, and that's really the mandatory condition to be able to do high resolution imaging, because those receptors are expressed in all cell types, not only neurons, but actually also many glial cells. If you are to label live receptors, endogenous receptors in a slice, you just don't see anything. It's way too dense. So the trick is really to be able to label a subpopulation of neurons. So you could do that by genetic tagging now with CRISPR. People are doing it. We started before CRISPR was really up and running. And so we did mice at the time, and with a very small tag, and you'll see it's extremely useful, and many people are using that. So the idea is to use an enzymatic method, and we are going to use the fact that biotin ligase biotinylates extremely specifically this lysine on this tag, and we are going to use that to make a knock-in of receptors tagged with this small tag, and then we are going to use a cell-specific expression of biotin ligase to have only a subtype of neurons being neurons where the receptors are biotinylated. So the principle is simple. We've done knock-ins, now we do CRISPR. It's easier to tag the receptor with this AP tag. And then we express B-ray as an endoplasmic reticulum resident protein. This is a technique developed by Alice Ting years ago. And the beauty of this system is that only you get cell-specific staining, but also you have the variety of tools which are extremely high affinity ligands for biotins. You have either monovalent avidin, which is a very small ligand, way smaller than a GFP or an antibody. We actually now have a variant of this monovalent avidin, which is in the nanomolar affinity, extremely useful, extremely clean to label slices. You'll see in a second. But we also use this tetravalent form of avidin to cross-link the receptors and immobilize them. So those ligands are relatively small. They penetrate extremely well in tissue. We use either the monovalent ligand or the tetravalent ligand. And as you've seen, we use the monovalent one to label receptors to track their mobility, and the tetravalent one to cross-link the receptors. We use knock-in animals, I told you, A1 or A2, and we express B-ray in a subset of neurons. And this way, we get very nice subpopulation of neurons labeled. This is one example of an organotypic slice with one cell only expressing B-ray. You see there's a sea of neurons, and only this one has receptors that can be biotinylated and are revealed by neutravidin staining. And you see this extremely high signal-to-noise ratio that allows you to do very nice imaging. So this was published a few years ago. And our next challenge was to be able to do single molecule imaging now with this tool in slices. Took us a bit of a time, but we managed to do that. These are still slightly preliminary data, but good enough that we have a paper in preparation now, which is really the first single molecule imaging, live imaging of receptors in brain slices. So this is a GFP expressing neuron in such an organotypic slice. This is GLUA2 labeling with this enhanced monomeric streptavidin. You see a very good signal-to-noise ratio, and you see maybe if the contrast is good enough, you see receptors diffusing there. The selectivity is extremely high. You are able to record tracks which are specific to the neuron that expresses B-ray. And if you do various types of controls, you have virtually no staining. So this tool is really highly selective and with a high enough contrast that you can actually detect single molecules. And that's thanks to using this light sheet microscope. We are in the beginning of the quantification, but something that was pretty striking when we looked at the diffusion rate of these receptors, endogenous receptors, I insist, in those slices, is that actually the mobility tends to be somewhat higher than what we see in cultured neuron, and we can discuss that. So this is work in progress, but I think it really allows us to re-insist on the fact that there's a large fraction of receptors which are mobile in synapses, probably about half of them. The other half is relatively stable in these nanodomains. And this exchange between mobile and immobile receptors and the exchange of mobile receptors within the PSD and with the PSD and with outside of the PSD, we think is very important for synaptic function. So I will end up with that, trying to keep in time. And the question is really, what is AMPA receptor diffusion trapping very good for? What we've done in the past, and I decided to remove the slides to make sure not to go too much over time, is showing that this diffusion trapping is extremely important for long-term potentiation. During long-term potentiation, you have diffusing receptors that get trapped, and this allows increasing receptor numbers in the postsynaptic density, and we've published that many, many times now. What I want to go to, which is newer in some sense, is this surprising but very robust role of surface diffusion in regulating short-term plasticity, and the form of short-term plasticity that's triggered by high-frequency stimulations. This is actually something that we discovered initially back 18 years ago, but that remained pretty silent for a long time. And now we've been revisiting that with, I must say, a better view, as you'll see in a second. So just a word on short-term plasticity. This type of high-frequency dependent short-term plasticity is the process through which, when you do a high frequency, meaning between 1 hertz and a kilohertz, stimulation of a neuron, presynaptic neuron, the postsynaptic response can either decrease or increase. You can either have facilitating synapses or depressing synapses, and even a mixture of the two processes. And these processes are thought to be extremely important for synaptic adaptation, but also for signal processing and information processing in the brain. As you all know, and still I would say more than 90% of the neuroscience population thinks that this is a hallmark of a presynaptic variation in transmitter release. And the fact that transmitter release is super important to regulate synaptic short-term plasticity holds true, of course. But one should not forget that there's a number of factors, of postsynaptic factors, that also regulate short-term plasticity. And of course, I'm going to highlight this one of surface mobility. So what we found nearly 18 years ago, nearly 20 was the experiments, was the work of Martin Heine, extremely talented thinker and postdoc in my lab at the time, who made this experiment where he cross-linked surface receptors in cultured neuron at the time, and he applied glutamate by iontophoresis or synaptic stimulation. And he made this extremely weird finding that by immobilizing receptors, he would see a very strong decrease of the second pulse of glutamate. And this was alleviated by cyclothiazide that prevents receptor desensitization. So we really scratched our heads for a long time. And Martin actually came up with this idea that it might be because the fast exchange of desensitized receptors might actually regulate synaptic transmission. So the idea is actually pretty simple. You have a normal synapse, receptors get activated very quickly within 100 microseconds. And then if receptors get desensitized, actually a lot of them get desensitized. And if receptors are immobile, if you have a second vesicle that gets released within the time frame of before the recovery of desensitization, then you have postsynaptic depression due to receptor desensitization. And what we postulated was that if receptors are mobile, then desensitized receptors can be exchanged by naive receptors, increasing the recovery from synaptic depression. And the very new data that we have now, and also I'm going to present some data from the lab of Ingo Greger, is that the reason why this is all working is that because indeed desensitized receptors do move way faster than naive ones. And we think that actually to keep a long story short, that even within every single release of glutamate, you have kind of a respiration of the whole PSD where receptors move faster than slower. So let me go through rapidly through the data that suggests that. So first of all, we showed that desensitized receptors diffuse out of synapses by applying glutamate on cultured neuron. You find a large increase in receptor mobility that's not observed in the presence of cyclothiazide to prevent receptor desensitization. And together with Françoise here in the audience, what we found is that desensitized mutant or non-desensitized mutant actually recapitulate this effect, whereby desensitized mutant move faster than naive ones. And so we came up with this hypothesis that actually desensitized mutant can actually unbind from auxiliary proteins, and this could accelerate their surface diffusion. And this is just an image of the extracellular domains of the receptors published initially for GLO2 by Eric Guo, but which apparently is not quite true, as you'll see in a second. And so this was confirming the idea that during paired pulse stimulation, you might have receptors moving fast enough to have desensitized receptors being replaced by naive ones. And now I actually got extremely excited by a new work by Ingo Greger, just published last year, and he was kind enough to give me a few slides, showing actually another mechanism that explains that, increased mobility, and I'm going to spend a few minutes going through that. What Ingo discovered is that in GLO2, there is a histidine here that interacts pretty strongly with a phenylalanine, which really blocks the subunits so that the homodimers of GLO2 are really rigid and they actually don't flip-flop like was initially proposed. And what he found is that actually this link is extremely sensitive to pH. And so what he published last year was that at pH 5.5, the extracellular domains actually are very floppy. They call that splaying, while at high pH, the domains are very rigid. And this is only true for GLO2, sorry. And so is this idea that the extracellular domains of GLO2 are splayed at low pH and are non-splayed at high pH. And he has all the molecular explanation for that. And so his hypothesis was that because during a vesicle release, you actually have a huge change in extracellular pH because there are hundreds of protons which are present in the vesicle due to the uptake of glutamate in the vesicles. This would be enough to have actually receptors playing and then maybe being unanchored from extracellular anchored and diffusing out. So he actually showed that. He did FRAP in various conditions. And you see that if you change the mutation in GLO2, receptors move faster. And if you look at different pHs, this is all consistent with the idea that GLO2 at low pH actually moves faster, and or GLO2 with a mutated histidine also moves faster. And so what Ingo has been proposing is that indeed during every single release of glutamate, you have those receptors which get desensitized actually move faster because yes, those splayed receptors are desensitized. So I'll finish just with a few slides of a paper that's in bioRxiv now, which is what are really the physiological consequences of that in the terms of the role of AMPA receptor diffusion in short-term plasticity and synaptic integration in intact tissue. So it took us a while to find the right preparation, but now that we had a knock-in mouse with this AP tag where we could investigate this study, Agatha and Angela in my group joined forces a few years ago, and we made this rationale decision that to have an effect of receptor diffusion on short-term plasticity, we have to work on a synapse where the probability of transmitter release is relatively high, and where the recovery from desensitization is pretty low, pretty slow, so that indeed in between two releases of glutamate, receptors don't have time to recover from desensitization, and that's where receptor mobility should play a very important role. So there was such a region, it's a S1 region of the cortex, and we started to do in vivo, recording in vivo injections of B-ray and recording is in acute slices. And we made trains, Agatha made trains of of stimulations, and indeed this synapse depresses, but when you cross-link the receptors to prevent their mobility, you have a strong increase in synaptic depression. We know this is synaptic because we measured glutamate release directly with iGlu sniffer, and you have no change when you cross-link receptors. And we also measured NMDA currents that shouldn't change if it's a postsynaptic modification, and there also it didn't change. Then finally, who cares about neutravidine? But we have a prediction, is that we've known for many, many years that synaptic activity, high synaptic activity such that occurs during long-term potentiation. When you do high-frequency stimulation, you induce massive calcium influx, activation of CAMK2, and we've known for more than 20 years that this triggers receptor immobilization. So our forecast was that during LTP, you might have a transient stage where receptors are immobilized with a change, and which this would induce a change in short-term plasticity. And indeed, that's what we found in these cortical L2-3 synapses. When you trigger LTP here, and when you look at short-term plasticity during LTP, you see this transient 10-15 minute depression, synaptic depression, short-term depression. That actually is something that everybody would have said is of presynaptic origin, and you can look in the paper. We have good evidence that this is indeed due to receptor immobilization. Before stopping, where are we going from there? I'm more and more excited about this role of synaptic mobility in looking at regulating short-term plasticity. And what we found is that this effect, although relatively modest, about 25% changes, has actually extremely large impacts on synaptic integration and network function. So just 2 experiments for that. First, looking at synaptic integration, meaning making trains of stimulation, bursts of about 10 action potentials, and looking at synaptic calcium influx in spines. You see that by blocking receptors diffusion, you have like a 50% decrease in integrated calcium in the spine because the effect accumulates over trains. And second, we also predicted that this would have a very strong impact on network coherence because of these cumulative effects. And indeed, that's what Angela found. These are organotypic slices, spontaneously oscillating, very correlated action potentials measured at the cell body level. And when you block receptor mobility, what you immediately see within seconds is that you get a decorrelated network property. And so we think that's very exciting. So I'm done. I will just maybe summarize this last part, saying that if you want to look at short-term plasticity, please don't only rely on the efficacy of transmitter release. You also have to consider receptor recovery from desensitization and receptor mobility. And it's really those 3 parameters that contribute to the construction of a short-term plasticity. And then finally, we have 2 experimental conditions where this is at play. Something we published a few years ago is that during LTD, you have actually an increase in receptor mobility that's translates into increased facilitation. And during early LTP, there is this receptor immobilization that triggers synaptic depression, and we think these are security mechanisms for the spine. So with that, I'm done. Just a few minutes late, please forgive me. These are the people who contributed to the work, the funders, and thank you very much for your attention.
With the mic, here it is.
Yeah.
Thank you very much, Daniele. It was an incredibly inspiring, a terrific lecture, and I think it was the most proper closure for our meeting. Thanks a lot again. Thank you. So maybe you can join me, and maybe now to close, there is a very important moment because we are going to announce all the awards of our Congress and the awards of our youth committee for the PhD meeting. So as first, I would like to call not only Danielle maybe, but also all the members of the local organizing committee, Paola Binda, Marco Norati, and Tommaso Pizzorusso, because they will help me in... Dov'è? Dov'è andato? Grazie. So, great lecture, really. So, maybe I can have my slides. That would be good. These are here, these are here. Oh, yeah, but I don't sit. I stand. Don't worry. A prossima. If we have the slides, we can announce the awardees. Maybe we don't have the slides. I don't know. Dov'è Andrea? Okay. It was very difficult, I've been told, to select the awardees. I personally was completely astonished by the quality of the research shown in these days. It was a great meeting. Thanks again to all, because the contribution from all our community made the Congress, definitely. So thank you so much. And here we are. So, Best Poster Awards. Ta-da. So as you know, we have 20. The first 10 ranked will get a little money prize plus 1 year free membership of our society. And the next 20, well, no money, but importantly, free registration for the membership in our society. So if you are in, come to the podium because we would like to shake your hands. So the first one is Fabrizio Ascone. Is Fabrizio around? No Fabrizio. I try with the second one. Marco Brondi. Marco, it's you? No, not even that. I'm sparing money, actually, for the society. Matteo Caldarelli, here you are. Come here. They will give you the awards. Congratulazioni. Matteo. Tu sei Fabrizio Ascone. Ah, we rescued the first one. Here, Fabrizio.
Congratulations.
Grazie, Fabrizio. Stay here, that we make a picture collective. Thank you. So, Matteo.
Okay.
Matteo Caldarelli. Matteo. One. Congratulazioni.
Lui è Marco Brondi.
No, we are not sparing money, Daniele. Marco Brondi. Oh, they were on top. Congratulazioni, Marco. Anch'io voglio congratulare. Thank you. We lost a slide, but I go by heart. Ilaria Cerchiaro. She's from my group. Bravissimi. Congratulations, brava Ilaria. Mattia Del Grosso. Lo abbiamo? È lì. We lost our slides. That's fine. Bravissimo. Complimenti. Tieni. Bravissima. Marina Di Domenico. Ah, wow. Oh, everybody is here. Ah, that's good. It's you. It's your turn.
Yes, it's always. It's a trick.
Complimenti. Spero di dirlo bene. Leiron Ferrarese. Non lo vedo. È nei palchetti, non lo so. Lo mettiamo qui. Wow. Arteda Pablecai. Here she comes. It's yours. Bravissima. Linda Scaramuzza. Bravissima, tieni. And then Erika Silvestri. Complimenti, Erika. Okay, now we start with the second part, but so it's great to have the prize. It's Carlotta Bon. Someone there. Bravissima, brava. Paolo Brunelli. Paolo Brunelli. Viviana Ciraci. Complimenti. Prendilo lavoro. Alessandra Cucinelli. What happened? Oh, the shoe. Okay. Bravissima. Eccoti. Silvia De Marchis. Ilaria Ghia. Eccola. Yeah, Congratulations. Maria Grazia Giuliano. Complimenti. Bravissima. Giusy Laudati. Yeah, now we go. Congratulations. Elisa Porchietto. Bravissima, complimenti. Manuela Tore. Complimenti. So now we have the awards that has. Now we have the awards. Now we have the awards that are given by the Youth Committee. So I would like to ask the Youth Committee to join me as well. There are a few prizes awarded. We have representative of our Youth Committee as well as the new president of the Youth Committee, Federica Campanelli, newly elected by them. Brava! You go.
So, for the best oral communication, Elena Ferretti. And the other winner for the best oral communication, Giacomo Turrini. Congratulations. For the best poster presentation, Erika Lucia Crapanzano. Congratulations. And the last, Eleonora Dall'Orto.
Thank you so much. Now, maybe we can call on stage, if I can have the next slide. There was another post, another awards. Exactly. We have, of course, we have all the travel awards that has been granted from our committee for the Congress. So there are many, many young people that were awarded of this travel grant. If you are here in the room, please come on stage, that we will have a very nice picture of everybody. Come, don't be shy. And there is another award that was generally given by Matteo Caleo Foundation that is been awarded to Giacomo Maddaloni. And again, if Giacomo is here, Giacomo, come all on stage. It's a nice group. Huh? Very nice. Yeah, we take a picture. It's nice. So, bravissimi. Everybody, this is the next generation of Italian neuroscience. Very proud of all of you. Please join us. So, now we take a picture. When you have a picture, somebody can take it. We have the official. Oh, there. We have the official photographer. Thanks a lot. And a very big thank you to our president.
Well, in this room there are several past presidents, but let me say, we never had one so efficient, very high power, very speedy president, and is Monica. So thanks, Monica, for all your effort.
Grazie. So we close, is the official closure. Thanks to everybody. It was fantastic. Grazie. Have a safe trip back. Oh. Wow. Grazie. No, gli sponsor li abbiamo anche detto all'inizio.
So, see you in Padua. Soon we'll have the dates.
No, dai. Grazie. No, facciamoci
Can I have a picture with Monica?
Yes,
Take one for you.
Yeah. Monica.
Lights. Please keep the lights on. We need to take a picture.
Okay.
Vieni, chiamiamo anche Fiorenzo, vieni, c'è anche Alessandro, facciamo una foto president. Non c'è, vieni su. Sì, facciamo tutti i presidenti che sono qui. Non c'è.
Cosa hai detto della... Micaela.
Michaela c'è.
Forse dovete fare un passo più avanti perché vi vedo poco alla luce. Ecco, così. Così mi sembrano più illuminati.
Una noi con Gaia. Gaia
la sto cercando.
Io vorrei una foto con il gruppo dei maschi. Guarda lui.
Va bene. Ciao, bravi tutti. Alla prossima. Ciao.
Ciao, grazie mille. Ci sentiamo.
Io l'ho detto all'inizio, ringraziamo, è sempre meglio fare all'inizio. Io per quello, che alla fine non sta Dov'è Andrea? Eccolo! Ne ha fatto
Guardare in alto.
Sì, questa volta diciamo che è stata impegnativa, però
Siete carinissimi, grazie. Ci hanno pensato solo loro,
No, grazie.
Loro personali.
Guarda, questi li dobbiamo tenere.
Spagnoli sono
Questo è mio e questi quelli che non c'erano.
No, infatti guarda, è un successo che non tutti quanti. Erano cresciute bene. Vabbè, no, che poi ho detto, vabbè, ormai concludo l'ultima slide. Sì. C'erano un paio di sinapsi, le iniziamo.
Però il delta ritmo c'era un po'.
Io voglio anche per tanti